Okay, Josh, I'm going to make you host for a second,
Okay.
and you see if what happens if I've got the YouTube running. We'll give it a minute, and then you can hand it back to me.
Okay. Do you want to leave the room and come back just to make sure that that works too?
Okay. It looks like it does end my recording when I do it, my local recording, which is not good. Okay.
Okay, sorry, I'm back. Let me make you host again. Oh, once you sign back in, you're host again.
Okay. All right. But the screen share died, so I have to redo the screen share. Okay. Okay. Mateo sends us a reminder that he's got to leave this afternoon. So we're going to have to be extra careful about doing the recordings today and tomorrow. Okay.
Okay, noted.
Hello, Lorenzo.
Hello.
Thank you for all the help. Appreciate it.
Oh, nothing. Trying to to do what I can. I'm recording all the things. I'm keeping them on my,
Great.
and I will share with you as when the thing finish.
Great. Okay. Is there anything we need to go over right now before we get as we queue up? Josh, Giuliano. I guess
No,
one
I'm
thing.
ready to present.
Right. One thing we're going to need to talk about is the hackathon. Who can help cover the hackathon? And then we also have to decide on that the rescheduling that we talked about today.
Mhm.
Sure. Oh, the breakout rooms. I need my own checklist for.
By the way, James, looks like Zoom stopped your recording again. Looking at the participants and you don't have the icon.
I don't know what that's about.
Yeah.
I can see the icon on James.
Yeah. Now it's.
I just restarted it.
Yeah. Maybe with cloud, when there is a hiccup, Zoom stops or something like that.
Well, that's another reason to try to do it with a. Oops. Shoot. Okay, I'll be right back. So it shows it is recording for me. Let me double check that the streaming is working. Josh, we forgot something important.
Yeah.
I was gonna say, I saw they put a new,
Go ahead, James. I'm sorry.
There was a request that we put the links for all of the demos that are going to be used today so people can download them now.
I did. They are uploaded. I was actually looking for the link. So we have the link to the folder directly.
Okay.
That's what I
so-
was working on right now.
Okay, but we should post that. We should put that in the chat now in the morning orientation session. And if we have a list of the link, if you have a list of the demos or not, I'll just put in a slide saying you're going to share that in the chat. Is that what you want to do?
Yeah, that we will share. I mean, the list is right there in the directory. They're all listed. But let me get the link for you.
Yeah, I've also uploaded my finished tumor sim.
Josh, can you put that link in the chat for me, or did you already do it?
Yeah, I did put it in the chat already.
Okay, let me find it.
Hello.
Hello.
Sorry.
Hello?
Hey, I will make you. Thank you for joining us. I'll give you the permission just a second.
Okay. Zlutka, are you there?
You want Jim Sluka? Did you say?
Yeah, I need someone to send an email to everybody so they know they need to download the demos.
Okay. I just posted that in the chat, but if you want to send him an email,
Right.
but send him an
Right.
email as well, that would be good.
I'll announce that in the Slack as well.
Great. Thank you. Okay, welcome everybody. We'll be getting started in just a minute or two. I know it's a long week, so I appreciate you coming back. Hope everybody got some rest yesterday and ready to go. Pedro, is Gilberto, do you know when he's going to be joining us?
Gilberto is not responding on WhatsApp, so don't know.
So,
He told me he would be here 10 minutes in 10 minutes, but he said that like 20 minutes ago.
well, if there's an internet problem, we'll find a way of handling it. We'll start in just a minute.
Good morning, everyone.
Oh,
Regarding this Compartmental version 4.2.5, I have a little question. Can someone help me in the breakup room?
let's see. What is it? Is it a operational question or what kind of question?
It is the thing is that in the console, for example, when it prints out the material in
Yes.
every example, in the older version, I see that whenever, for example, in the steppable, I print out something, it shows up on the console. But on the Windows, I don't see in the output. So I'm not sure if there is some problem in the installation or it's my problem.
That may be a change in the functionality of CompuCell in the latest release. The printing actually is fairly slow, so it may have been disabled. We'll have to wait for TJ to answer that question.
Oh, okay.
Unless Giuliano, do you have an answer for that?
I couldn't hear the whole question. Could you say that again?
The question is that in the console part, when it prints out the the things,
Yep.
it doesn't show up. For example, if I print something in the steppable file, so it doesn't show up in the console part. I mean, I'm talking about the pie chart console part. So
pie chart?
yeah, if I can share a screen in the breakout room if you want, we can go through that.
Oh, well, okay.
Yeah, I mean, it could be a configuration on PyCharm, but I think I've had issues with printing before where it didn't actually print sometimes.
Yeah, in the previous versions, it is possible, but I checked it with different computers, and so it didn't help.
And when not using PyCharm, does the print work normally?
I think in the player, you mean?
Yeah.
In the player, I think no. In the player, I haven't seen that it prints out.
Okay, so, sorry, let's hold on for just a sec on that. We'll hook you up with TJ when he gets on.
Okay.
TJ, let me get started, and then there's a technical question. If you can answer it, either depending on the time, either in the session or in a breakout room. Apologize, but let me just get formally started, and then we'll go from there. Okay, welcome everybody. Thank you all for coming back to day 3 of the workshop. Today we're going to talk about cell growth, cell division. We'll build a very simple model of a avascular tumor where the cells take up nutrient from the environment and grow in response to the nutrient. That's actually a pretty good basis for learning how to do much more complex models. We're going to have a discussion of Chemotaxis and a demonstration of how to use that to do a simple model of actin polymerization and cell crawling. And then we're going to do a short introduction to network modeling inside of CompuCell. So it's a lot of material to cover today. As always, these sessions are being live streamed on YouTube and are recorded, so please be aware of that. I think people have managed the Zoom logistics by now. If we have any problems, please let us know. We'll be doing some polling, so that's also going to be something that you'll see. I'm going to wait for the main session and do a quick poll on the hackathon. There was a very good suggestion yesterday that we should try to make as many of the demos as possible available for people to download. If you would like to do that, we have put the link to the downloads for the demos. Can't promise they're all there, but mostly they should be there. So if you want to take the time between now and the start of the main meeting to download the demos to your local machine. And if you're using NanoHub, you might want to use Jupyter to upload those demos as well. We will go through the uploads and downloads as we need to do it, but if people have got that set up beforehand, it will save time and probably some frustration. So again, today, cell properties and behaviors, cell division, avascular tumor simulation. Gilberto will be presenting his very nice demonstrations of cell migration. Primarily, we'll be building together a 2D cell migration demonstration, and then Gilberto will share and show you a 3D simulation using compartmental cells. Those are a little bit more complicated, so we won't be able to build it together, but it's a great framework to build off of. I want to call your attention to tomorrow in our second module, which will be devoted to hackathon work, and then the additional time we'll be using on how to load externally defined SBML models into CompuCell and run them. We had some great presentations of hackathon ideas yesterday. People who are interested in pursuing the hackathon but haven't got their own projects, and I'm going to do a quick poll on that now. I'd encourage you to please contact the people who are proposing projects. Again, we can reshare. Maybe Giuliano, you can reshare the links for the Slack channel for the hackathon, the Google Doc for the hackathon, and also the folder of hackathon ideas. There were 3 people who were not able to present yesterday. They're going to planning on presenting their 3 ideas tomorrow. Again, these hackathons tend to work best if we have teams. So don't be shy about asking to join a project that you find interesting. Nobody's required to, if you have your project and you have your team already, you can certainly say no to people joining. But I encourage you to take advantage of expertise if it's offered. On the hackathon, I would encourage people also. Okay, so the schedule today, sorry again, is cell properties, behaviors, and cell division by Josh. The avascular tumor simulation using those things by Giuliano. Gilberto's talk on cell migration and chemotaxis. And then I'll be introducing the network models at the end of the day today. Again, the hackathon will be Saturday and Sunday. Please work as much as you can to self-organize teams. We'll do our best to help out, but really the hackathon philosophy is self-organization. So please do that. And we would, I'd ask you to think about, especially for people who haven't done this kind of modeling before, do take some time, not that you have a lot of time outside of these classes, but take a lot of time to try to develop your hackathon ideas. In particular, I think it's important to try to identify what it is that you want to predict and be as specific as possible. I want to know the number of cells in the tumor as a function of time. I want to know how many cells differentiate. I want to know how big the rosettes are as a function of changing the contractility of the apical ring in my epithelium. Try to be as specific as possible, and then ask the question, what do you think you need to build a minimal model? Our hackathon is only going to be 2 days this year. That's actually a lot of effort from the people participating, but it's also not a lot of time. So you should try to be realistic about how complex a model you can build in those 2 days. Focus on trying to get something that can be a base model to be extended. And so one of the things to start thinking about is what are the minimal pieces that you need to build that model? What are the components? What kinds of cells? What do the cells have to do? How many cells do you need in the simulation? How complex does the model need to be? And of course, that's not so easy to do if you haven't worked with this kind of modeling before. But the more you can think about that, the better. Again, please try to see what you can do in terms of teaming. We're going to have the second session tomorrow open for discussion and teaming. If you'd like a breakout room in that session to have conversations, let us know. We can have an open pit discussion for a while, but probably teams will want to get together. And so if you let us know that you want a breakout room, we'll create one and allow people to move around that way. It's not that Zoom isn't the ideal way of doing that, but it's not a bad way to do it. So again, for this morning, I would encourage people to try to download the demos. If anybody has trouble with that, please let us know, and we'll do our best to help out. And then upload those files to the NanoHub Jupyter Notebooks, using Jupyter Notebooks or Tellurium. If you're going to run them on your local machine, once you've got it downloaded, you're okay with that. So those were the things that I wanted to cover just as introduction this morning. There were a couple of questions. There are 4 people who said that they need to present their project ideas for the hackathon. I knew 3 people who said that they needed to do so, but if there's a 4th person, please reach out to me and let me know if you upload your slide or how you want to present, and I'll do my best to help out.
Oh, it's me, and you can ignore it because I misunderstood the poll.
Oh, okay, fine. No worries. I just want to make sure that everybody has the opportunity to present. I know 3 people were unable to do that yesterday. Okay, so with that, we can answer other questions. Are there any questions? There's a question, can I submit my hackathon slide now? if yes, so there is a link. Giuliano, could you share the link to put the hackathon slide up? The Zoom link for the Zoom link, the Google Drive link for, and the Google Doc. We will repost that material. There were some other questions. There was a question, can we implement cell polarity in the Hamiltonian so that there is an energy that is dependent on the dot product of the cell polarity and some external vector? The answer is that you can in fact implement any additional energy functionals, what are called plugins in Compucell, using the C++ extensions. And there is some documentation for that online. It's not extremely difficult, but it is a little bit more involved than doing a Python or CC3D-ML scripting. And if you're interested in learning about that, I would reach out to TJ in particular. Giuliano also knows how to do plugin development. The key thing always with doing a plugin is to define what it is you're trying to accomplish with it, and make sure that it's designed in a way that's generally useful. We're delighted when people want to develop plugins. The documentation, there is a developer's manual on the Compucell manuals page, but please do connect to TJ and Giuliano for the details on that. Were people able to find the links? I will post the links one more time for the Google Doc, for the hackathon. So the Google Doc for the hackathon is here. Okay. And to upload to the hackathon slides. The directory is here. And please remind me, send me an email if you want. Make sure that you get included among the tomorrow so that you can present. So again, there's no requirement that people who've already present anymore, but I do want people to have the opportunity to discuss their projects, either as a group or in breakout. I'll try to join you on that. So are there any other questions in the 5 minutes we have remaining before we get started? I'm looking in the chat to see if there are any questions. There was a technical question about whether the yes, the link for the slide is what I just pasted. The slides. I guess the question, somebody said, okay, so there was a request for people to please put their lectures, the draft lectures for 3.1 and 3.2 into the student materials folders. Josh, could you take a minute to save your lecture?
Yes, I thought I have uploaded it, but I'll do it again.
Let me check. It's there. It's there. I see it. 3.1 is there. So if the person, Aaron, if you could check again, it looks to me like 3.1, 3.2, 3.3, and 3.4 should all be in the student materials. I know Google Drive doesn't always sync instantaneously, so it can sometimes take some time. And of course, as we work through the day, we sometimes, or afterwards even, we sometimes catch mistakes and improve the slide deck. So please be patient if the slide deck that you get isn't exactly what shows up during the lecture. And we'll continue to work to try to make the video materials available online. Please send the link to the online videos. So there are two sets of online videos. Giuliano, can you do that? In the student materials folder, there's a directory called videos that should have versions of all the modules, or at least most of them. There also are the YouTube the YouTube recordings of this. The YouTube recordings are complete, but they're not the most convenient thing in the world because they're the entire monolithic 7 hours. You want the live stream? Okay. Let me give you the live stream. Okay, got that. Any other logistical questions or issues before we get started? Were people able to find the link for the demos and do the downloads? Again, that was a very good suggestion to try to get that done. We won't assume that people have done it, so we'll walk through that again. But certainly make your life a little easier if you have that in advance. Okay, thank you. Yes, you should. If you're using NanoHub, please do upload the demos to your NanoHub account using Jupyter. That'll save a lot of headache later on. Okay. Any other questions? Okay, well, in that case, I will turn it over to Josh. Do you want to screen share and probably wait till 10:33 to start, but you can put your slides up, set up.
Okay, sure. Again, for everyone who's already joined, I would highly recommend downloading the demos from the link I'm posting in the chat right now and upload them to NanoHub if you're using NanoHub, or at least also unzip them locally if you're using your local version. And we'll start in about 1 or 2 more minutes. I'll give that time for everyone to catch up. Okay, hello everyone. Welcome to this third day of the CompuSol 3D workshop. Today I will be presenting the module 3.1, introducing cell properties and how to tie some of these properties to cell fields, to chemical fields. Just as a reminder, this presentation will be recorded and live streamed. Again, I highly recommend everyone to download the demos and upload them to NanoHub if that's a platform that you're using, and unzip them locally if you're using computers on your local machines. So as I mentioned, we'll be talking today about cell properties, in particular cell division, how to implement cell division, cell growth, and cell division in CompuSol, and how to tie some of these properties to the behaviors of the chemical field. In the first part of the module, I will be talking about how we can think of cell proliferation from a modeling perspective, how we implement it in CompuSol, and what are the considerations that we have to take when we are making our cells grow and divide. So the basic principle of life really is that cells have to undergo proliferation and division. And in cellular organisms, it's particularly important for cells to decide when to proliferate, when to survive, and when to die. And these decisions are made based on signals that the cells receive both internally and externally. And there are factors such as cell contact or factors such as cell-to-cell contact that might inhibit this cell growth and proliferation, and other factors such as growth molecules that might induce cells to proliferate. And we will see examples of modeling these two types of behavior. From a biological standpoint, when cells undergo proliferation, they usually start by doubling the material, the DNA material in the cell. And once they have doubled this in this S phase, when they have doubled genetic material, they undergo actual cell division. And then after they remain in a rest state where they decide, or they cross certain checkpoints where they decide whether they want to continue proliferating or they want to arrest cell division. And there are multiple of these checkpoints along the different stages of the cell cycle. I'm not going to go into the details of these because from a CompuCell perspective, we are not going to model the cell cycle in detail yet. And I said yet because tomorrow we will see how we can couple the cell division that we will be discussing today with internal models of the cell cycle, with internal models of the cell cycle that might control how the cell divides. But at least for the purpose of this module, the basic idea behind cell division is when we implement it in CompuCell is that we will make our cells grow, and they will divide when they reach a particular size. And I know, again, I'm sorry that my screen is a little bit small. I am, it's because I have this split screen, so because I want to be able to share my code and my presentation. But the slides are also shared on the Google Drive. If you want to have it as a reference, so you can see it side by side while I'm talking, you can also download them. Juliano, if you can post the link to the directory with the slide decks. So coming back to the idea, or summarizing here, the factor and processes leading to cell division and proliferation are very complicated in the real world biology. For the purpose of this module, we will simplify all of that complexity by modeling cell division and we will simplify cell division by having our modeling as our cells growing and as our cells dividing once they reach a particular size. So how do we make our cells grow? So in CompuCell, the simplest way we can make our cells grow is to increase the target volume of the cell by a certain amount each time step. How we implement this cell growth can be as complicated as we want, and we will see different ways in which we can modulate cell growth. But again, the idea is that the idea will be that we will increase the target volume of the cell, which represents a increase in the mass of the actual cell. And then once we have reached a particular size, then we will have that cell divide. Another important thing to notice here is that we actually do not increase the actual volume of the cell. What we increase is the target volume, and the actual volume will be the result of the interaction between the different energy or constraint terms that we include in our simulation. So we can set ourselves to, we can set the target volume or increase the target volume constantly, but the actual cell volume of the cell, whether it reaches that target volume or not, will depend on other factors. So for example, whether the cell has space or not to do it, or whether we adapt what the surface area of the cell should be. And all of those factors will play together to determine what the actual volume of the cell is. But in principle, the best way we have to control whether we want the cell to grow is to increase its target volume. Now, and remember that increasing target volume the target volume acts as a spring force in our energy, in our energy terms, that basically forces the cell to increase. Another thing that is important to notice is that we cannot define, so far we have been doing a lot of modeling from the XML, but if we want to be able to increase the target volume of our cells, we have to do it dynamically from the Python script, and we will have to define it inside the Python script, and we'll have to disable any specification of the target volume in the XML. Otherwise, we wouldn't be able so the first exercise, this is exercise 311 in the demos that I shared. What we will do is that we will create a simulation domain. We will have a single cell, and we will assign contact energies, and we will assign the volume constraint. And we will show how to select a growth steppable from the wizard, so that facilitates us implementing cell growth later on. Then we will initialize a single cell in the center of the simulation domain, and then we will have this cell growth by setting a growth rate of 25/500. And then we will, after having observed growth just with the volume constraint, we'll add a surface constraint and see how the surface constraints affects the cell growth. Okay, so first, to start the let's start creating the simulation. So we create a new project. I'm gonna click growth here. And then, I'm sorry. And then I'm gonna create a 100 by 100 lattice. Now I do have cells that move, so I'm gonna set my boundaries to be periodic. I don't want the cell to bounce against the borders of the simulation. We talked about what periodic boundary conditions meant yesterday. If anyone wants a reminder, I would direct them to the presentations yesterday, but the same ideas that apply to the field apply here to the simulation. I'm gonna use the blob initializer. I'm gonna create one cell type called cell. And I'm not gonna add any chemical fields. And now I'm gonna add the contact energy constraint, a growth steppable, and the volume local flex. Oh yeah, I'm sorry. So I did mean to change it, the pixel copy range to 3. Thank you so much for the reminder. Yep. Okay. And once I've selected all of the properties that I want to add, then I can finish my, with the I can finish, I can generate the simulation. So once I've generated the simulation, the first thing I should notice, yes, please, I forgot to mention it. See, if you didn't, you can mark the surface local flex. If you didn't, I'll show you how you can add it later on, because if you forget to add some of the properties, you can always use the pull down menus in Intuitive to add the steppables or the plugins that you might need to add. So if I go to the XML on this file, I would notice that I've defined my simulation domain. One second. And I have 2 cell types. And notice that I now have this surface and volume plugins, and that they do not have any information. Now, let's say that we forgot to add the surface plugin. I can always come to CC3DML, plugins, and then I hit and surface. Right? And then it will paste a snippet with the information to specify the surface plugin. However, we want to be able to specify the parameters of the surface constraint in the Python's steppable. So we will have, we want to delete, get rid, or comment out the information inside the plugin in the XML file. And then we will also notice that I have now the contact energies. We're not gonna play with these contact energies now. And then that I have a blob initializer, and I've set the blob initializer so that I initialize one single cell in the center of the simulation domain. So I just set the radius to 5 and the width of the cell to 5. Okay, now I said that we have loaded the surface and the volume plugins, but we haven't specified what the properties of these constraints or the parameters of these constraints are, because we will do it in the Python step. So if you don't have the blob initializer, you can again always come to CC3DML steppables, and then yeah, okay, then you can delete the information in the blob initializer, come to CC3DML steppables, and then you can click on blob initializer, and that's gonna add this code snippet here, and then you can modify it as in the yellow lines in the okay. So now if we come to the step, as I was mentioning, if we come to the steppable, we will see that that we have specified that we now have to specify what the cell target volume and the lambda volume is. And then we will see that we also have this gross steppable that allow us to increase what the target volume of the cell is. So all I need to do for this particular simulation is to set the target volume, and since this is happening at the start function, this is gonna be the initial target volume. So I just have to set that one to 25. I need to set my lambda volume to 3. And then in the growth steppable, I will increase my target volume by the rate 25/100, which means that every 100 Monte Carlo, the cell is gonna grow 25 pixels every 100 Monte Carlo steps. Okay. So I'm gonna wait for everyone to catch up here before running the simulation. Okay, good. Looks like everyone is with me. So now if we execute this simulation, what we see is that as the simulation times goes on, my cell increases because I am increasing my target volume. Okay. So I'm gonna stop this simulation here now and continue talking about how I could add more constraints to this simulation. I'm sorry, there's a couple of questions. Let me come back to that. It's not, right. It doesn't happen every 100 steps, right? It continuously, so it basically increases 0.25, the target volume, every Monte Carlo step. So I can show the XML again, but I didn't do anything fancy. I just initialize the blob initializer with a radius 5 and a width 5. That's gonna give me a single cell in this center of the simulation. Radius means the radius of the blob, as people are discussing, not the radius of the cell. Width is the length of the cell. Okay, so now we can add an additional, because we have the, in the XML, we also have the surface constraint or the surface plugin loaded. We can also specify an additional constraint in our cells, and that would be a target surface. And by surface, we mean the number of pixels that are the boundary of the cell. And again, since I've commented out that information in the XML because I want to be able to modify it on the fly. I mean, as at a constant rate, I can specify it here in my steppable just below what I've specified the target volume and lambda volume. And it will be target surface and lambda surface. Okay. So I'm just gonna do that now. So I'm basically adding a target volume, a lambda volume, a target surface, and a lambda surface. Oh yeah, there is a typo in the slide. I'm sorry. It should be lambda surface on the last line here. I'm sorry about that. Thank you for catching that up. So quick question, what do you think it's gonna happen once I run this simulation now, right? Because my target volume, it's increasing, but the target surface is not, right? I'm not readapting, right? So the surface area will constrain what the volume increase will be. So let's run, now that we have, let me make sure that everyone is with me. So just as a recap, I first initialized the volume constraint, and I am increasing the target volume by a particular rate every Monte Carlo step. And now I am adding an additional constraint, which is the surface constraint. And now I can execute my simulation again. And as some people mentioned in the chat, the surface constraint is now constraining the volume. The cell is very squared because I'm basically adding more and more target. I'm increasing my target volume, but I'm not readjusting my target surface. So it basically wants to increase. It wants to increase the target volume, but the target surface remains the same. Good question. Why is it still increasing even if I am not increasing the surface area? Well, because or the target surface, and the reason it's because there is always a interplay between the different constraints. It's not like one constraint totally inhibits the other, right? So what's happening is that the strength of these constraints are similar, and the magnitude of these constraints are similar. So the cell is still trying to achieve or meet the both of the constraints at the same time. So it still increases the area, but it's not. It still increases the area, but it has what we'll see later. We'll call later. It's under a lot of pressure. We'll come to that idea later, and we'll see how that. And that's why the cell it looks very square and not very motile in this particular simulation. But the idea here is basically that we are putting too much air into a balloon, or too much water into a balloon that is relatively small, and it's very, very tight. It's very under a lot of pressure, but that prevents the cell from moving. If I wanted to, for example, if I increase the lambda surface by a big factor, so I make the importance of the lambda surface much bigger than that of the, I'm sorry, if I make the importance of the surface much bigger than that of the volume, then in that case, I think it was Polly who had that question. Yeah, it's not growing anymore because now the surface constraint is much important than the volume constraint. So the cell will try to meet the surface constraint rather than the volume constraint. Okay. So let's put back this to 3. So we're basically making them the same, about the same magnitude. So the way we can solve this, if we have these two constraints, if they have the interplay between these two constraints, is to adjust the target surface of my cell as I increase the target volume. So yeah, that's what you see. So the way I can adjust the surface as a function of my target volume, there's many ways you can do this, but the easiest way is to assume, for example, that my cells should occupy the area of a circle. And then given the new target volume, I can infer what the target surface would be from that target volume. I think it's divided by 2 here, but just as an approximation. And then I can readjust my target volume, my target surface. I'm sorry, this doesn't work here. I can readjust my target surface every time I increase my target volume, such that the target surface readjusts itself. So I'm gonna relaunch the poll just to make sure. I'm sorry. I'm sorry if I take a little bit of time. I'm just trying to keep up with the chat to see which questions I can answer and which I cannot. So the reason it's pi in this particular simulation is because I've already, and I forgot to mention this, thank you for the question. I've imported pi from the math library. So it's available to me. And let me relaunch my code. Let me come back to and relaunch my code. So all I did was, after adding my targets, my surface constraint, I'm modifying the surface the same way as I adjust the target volume. Okay, good. Everyone is with me. So now when I execute this simulation, sorry about that, I notched. So the cell starts growing too, and I said that I have a factor of 2 missing somewhere. That's why it looks a little bit squidgy in the edges, and that's because I have my target surface is just a bit bigger than my target volume. Okay, but now I can meet both constraints at the same time, or my cell can meet both constraints at the same time, because as the cell increase, as the cell volume increases, I readjust what the target surface should be. Or as the volume increases, I readjust what the target surface should be. Yes, AJ, I'm sorry. For those to import Pi, there's many ways, many libraries you can import it from, but probably the easiest one is, sorry. From math import Pi. Okay. So I have this version of the code in exercise 311. Some of the additions are commented out in the original version, but you can play with the different combinations of the constraints and how to modify them. So now that we have make our cells grow, now we can start thinking about how to trigger cell division. It's really, there is no predefined condition to in Compucell to trigger cell division. It's up to the user to define what condition will trigger the cell division. As I mentioned at the beginning, in real life, there's many conditions cells have to meet in order to be able to divide. They have to have the right signals. They have to have enough nutrients. They have to have enough space. They have to double their genetic material, and then they have to undergo mitosis. In Compucell, as James mentioned at the very beginning, this is nothing happens unless nothing exists, and nothing happens unless I explicitly state it. So I have to state what the conditions are for our cells to divide, and I have to be I have to make sure that the cells are able to meet those conditions if I want them to divide. And once they've met those conditions, then there's other options or other factors that I have to take into consideration in terms of what happens during and after division. So what plane the cell's gonna divide, I have to be able to specify that, and whether after division the cell both daughter cells have the same side, or there's a cell that is bigger than the other. Normally we think of cells as dividing into two symmetric cell division, where cells divide into two cells of the same size, but that is not always the case, especially during development. So again, the simplest condition that we can think of, or we can implement in Compucell, is to have our cells divide once they have doubled their initial volume. And once they have divided, then we have to have the daughter cells inherit modified versions of the target volume and the target surface of the mother cell, right? If you imagine that the target volume represents the mass of the cell, then you should expect in symmetric division, you should expect the daughter cells to inherit half and half of the volume of the volume of the mother cell. When implementing cell division in Compucell, there is this special step I will call mitosis step that has a little bit of a different structure, and has it has all the available options that you will need to implement cell division. In particular, it will have it will ask you to specify what the condition is and what happens to the cells after they divide. And just as a teaser here, the mitosis step has this structure where in the step function we have this cells to divide dictionary, and once we specify what the condition for cell division is, we append the cells that we want to divide to this, it's not a dictionary, I'm sorry, it's a list. We append the cells to that list, And then we iterate through this list and perform cell division on those cells. And then we can divide them along random axis, or we can actually specify what axis we want the division to occur. And then we have this special function called update attributes, where we decide what happens to the attributes of the cells once they divide. And the way to specify, the easiest way or the simplest way to specify is to clone the pattern properties to the child, but we want to divide the target volume of the cell in half, or the target volumes of the daughter cells in half, such that the mass of, such that we divide the mass of the mother cell into the daughter cells. And there is more control options that we have, but we won't go into those right now. We'll see those in later in the exercise. Okay, so let's go ahead and start. Let's I'm sorry, I deleted one slide that I wasn't meaning to, but what we will do is that we will create for this exercise, we'll create a simulation domain, and we will add two different cell types, stem and terminally differentiated. And we will add constant volume, growth, and division from the cell properties. And we will initialize a single stem cell in the center of the simulation domain. So let's do that together. New project, I'm gonna call it division. Again, 100 by 100. And I keep doing 100 by 100, but again, this is up to you whether you want more or less resolution or more or less space. We're gonna have periodic conditions on the X and Y axis, the blob initializer. We will add the stem cells and the terminally differentiated cells. No chemical fields. And then we will choose, again, yeah, sorry, pixel copy range 3. It's always good to avoid lattice effects. And then from the cell properties and behaviors, we're gonna choose contact, growth, mitosis, and volume local flex. I'm gonna wait for everyone here. Just 1 second. So you can specify it if you're interested in adding a surface constraint. I'm not gonna be, just for simplicity, I'm not gonna be adding the surface constraint again in the exercise of this module. But if you want to add that surface constraint for any particular reason, depending on the model of interest, you could also add it. But we won't for the purpose of this module. Okay, and then we hit next, and we finish, and we open the simulation. What I'll do is I'll open instead of typing in everything from scratch. I'm just gonna open the exercise 312, which has the code for this exercise. Okay. And then we can see that we have created two cell types. We have the contact energies between them. We're initializing a single stem cell in the center of the simulation domain in the blob initializer. So if you're typing this from scratch, make sure you adjust that. So always use a radius of 5 and a width of 5 to ensure that I have one single cell in the center of the simulation domain. And that my boundaries are periodic. Okay. So the reason I have one type of cell is because I'm gonna start with one single cell, and I want it to be of the stem cells for the later on the exercises. I want to start with the stem cell for later on the exercise, so I just went ahead and set it here. If I said stem and terminally differentiated, it will randomly choose which one of them to start with. But I always wanted to start with the stem cell, so I just set the type of the cell I'm initializing to be a stem cell. Now, if we come to the steppable files, there's more stuff that I added here. But if you start from scratch, what you'll have is a constraint initializer with the target volume and lambda volume. You will have a growth steppable. I'm sorry, this is moving way too fast. Where the target volume of the cell is being updated. And you'll have the mitosis steppable that we talked about earlier that has the cells to divide list, the condition for when that division occurs, the implementation of the division along a random orientation, and then the updates attributes, and then the updates attributes of a special function. Okay. Now, if I run this simulation, so note here in this part of the code that I am actually changing the type of one of my daughter cells. So if the parent cell, let's say, is stem cell, then the daughter cell will be, one of the daughter cells will be stem cell, and one of the daughter cells will be terminally differentiated. So if we run that simulation now, let me make sure that everyone with me before I run the simulation. So again, here, this part of the code is determining that one of the daughter cells is gonna be of type 1, which is my stem cell, and one of the daughter cells is gonna be of type 2, which is the terminally differentiated. Okay, seems that most people are with me, so I'm gonna go ahead and open that simulation in Player. I'm not gonna use this now. So as I mentioned, we start with a single cell in the center of the simulation domain. So this, Polly, you said this part is missing for you. It's fine for now because we won't do anything with it right now. But we'll come back to that a little bit later during the exercise, and it's not that complicated, so you can type it directly if you're missing it. But now for this part of the exercise, Okay. It crashed. 1 second. I'm gonna have to relaunch Compartmental here. Did it crash for anyone else when they try running it? That's exercise 312. Okay, it ran for you. Okay. Okay, I'm gonna minimize this. So what I can see is that my cells are growing and they are dividing, and then I have cells of both types. In this case, blue is the stem cell and green is the terminally differentiated. And the reason I have cells of both types is because when my cells grow and divide, one of the daughter cells is going to be of one of the types and one of the other one is going to be of the other. So should have about the same number of cells for each type in the simulation. And I'm going to start it over. And if I let it run for long enough, the cells are just going to fill up the whole simulation domain and they'll stop growing and dividing because they have no additional space to do so, and we'll come back to this idea later. Okay, so yeah, Ying, we'll come to that idea later. That's why it's added. That's why the plot is added in the code, because we'll discuss what's happening. So now we can try different conditions. We can try dividing it. I'm gonna leave this to you, but you can just change what the condition is, and you can tell. So for example, instead of doubling when it reaches a target volume of 50, we can make them divide when they reach a target volume of 30, or we can also divide them along different orientations. Okay, but I will leave this part of the exercise for you to do as a take home, so you can explore what the effects of these of changing the conditions are. So now, so how to control the rate of division? So the rate of division I can control by changing what the rate of growth is, right? Because that's the only condition that we are implementing for our cells to undergo cell division. And I do so in the in the part when I'm modifying the growth rate, I can make it grow slower. Sorry. So in the part when I'm making our cells grow, instead of 25/100, if I do it 25/500, then that will control how fast cells are growing and consequently how fast they are dividing. Okay. Now I want to, instead of having all of our cells grow and divide, I just want my stem cell type to grow and divide. And once they are in the terminally differentiated cell, I want them to not grow and not divide anymore. So again, there's multiple ways I could implement this, but the simplest way is to modify which cells I'm actually increasing the target volume. So if we come to the code in line 44 here, in line 43, you see that I'm iterating over in the growth step. I'm iterating inside over all of my cells, which is my cell list. and I'm increasing the target volume of all of my cells. But if instead of doing that, I iterate over my stem cells and increase the target volume, then the stem cells are the only cells that are gonna grow and the only cells that are gonna divide. And to do that, I have this syntax here for the cell list by type that I can iterate over. And if I iterate only over the stem cells, then I can just increase the target volume of the stem cells. So I'm just gonna comment out this part of the code and uncomment this part of the code and launch my poll. Just to make sure that everyone is with me. Okay, everything I did here was there's some people that are still working on it. So the only thing I did, right, for the purpose of this part of the exercise, all I want to do is I want to have the stem cells grow and divide. And the way I do it is by, instead of in the growth step, instead of iterating over my whole set list, I iterate over the set list by type. And the type I've iterated over is the stem cell type. And see how I reference the type is by referencing self.st, which is my stem cell. And those are the only cells that are gonna grow, and consequently are the only cells that are gonna be able to divide. So let's run the simulation again. So now you can see that only my stem cell is doubling in size and dividing. Once it's divided into a terminally differentiated cell, that cell doesn't increase in size, so it doesn't divide. So now I've shown you how you can have better control over right. I mean, as I mentioned, Polly, there's multiple ways you can try to implement this. I chose to do it by just having the stem cell divide. But I could also have, for example, all of the cell growth and only my stem cell divide. I could do so because in the mitosis step, I also iterate over my whole cell list. If I were to iterate over the cell list by type, then only those cells will be appended to the list that I'm using to divide cells. I don't know if that makes sense to you, but right. So the condition, it will change, and again, but then what will happen in this particular case is that all of my cells will grow, but only my stem cell will be the one dividing. And you can try that yourself as a homework too, if you want to. Okay, I'm gonna continue moving on because there's other things I would like to cover. So as some people notice, this code already had a, and that's because I forgot to delete that plotting code from this exercise. But this exercise already had a code where I was plotting the average volume of the cells in my simulation. And what some people noticed was that as when the cells occupy the full space of the simulation, domain, that the average volume, the average target volume will continue to increase. And the reason that it's happening is because that we have no way of, we haven't specified any way of the growth rate is always constant, right? And the only way that the target volume decreases is when the cells divide. But if the cell cannot divide because it doesn't have space, then the target volume will just continue to increase. That's what we call the cell. That's when we call our cells to be under a lot of pressure. And pressure is just the difference between what the target volume of my cell is and the actual volume that the cell wants, that the cell has. So if the target volume is much bigger than the actual volume, then we say that our cells are under high positive pressure. Okay, and there is a derivation that we can do from the energy term to the pressure. But in particular, but we won't get into the details of that. What is important to notice for the purpose of the module is that the pressure of our cells is proportional to the difference between the target volume and the actual volume. And if there's no more space for our cells to grow, they won't divide, and the target volume will continue to increase, and so will do the pressure, or our cells will be under a lot of pressure. If we wanted to avoid putting ourselves into a lot of pressure, we could modify our growth rate such that instead of being a constant growth rate, the growth rate is a function of the pressure cells are under, and there is multiple functional forms that this modification could have, depending on how smooth you want your transition to be between, let's say, cells, between when the target's volume stops increasing. So now we will, the advantage of using this modification of the growth rate due to the pressure is that it allows us to implicitly model contact inhibited growth, right? In the context of cells that are growing and fill up a full space, if they don't have space, to divide into, they stop growing, right? And cells live happy in those conditions. They're not inflated balloons. So we will want to, if we want to mimic that, we want to reduce the pressure our cells are when they occupy the full simulation domain. So we will do that by modifying the growth rate using the pressure of the cell as a reference. I'm gonna walk through this. So the first part of the exercise is basically using the same basic simulation specifications as in the previous exercise, but we will add additional information into that one. So I'm not gonna create a full simulation from scratch. Well, what I'll do is that first I will add a plot that shows the average volume of our cells. And just as a quick reminder, if I want to add this is inside this 4-cell loop, so it's creating a lot of, let me move this out and move this out. So, so just as a quick reminder, if we want to create a plot window, we go to a start function. It can be whatever start function we want. And then we go to CC3D Python scientific plots setup. And that's gonna give us a code snippet that we can modify to include, I'm sorry, here it moves really fast and no, when I scroll, that we can modify for the specific details of the plot that we want to add. Let me decrease a little bit. So we're gonna create a plot window that has as a title average target volume, and we're gonna create a data series called AVOL. And I'm gonna wait for people to do that. I'm gonna give people a couple of minutes to be able to do that. Please make sure that the indentation is correct and that the plot window is not being created inside the cell build. I'm gonna relaunch to give everyone some time to create the plot window. So basically all we did was created a plot window by going to CC3D Python, scientific plots, and setup. And once we had the cut snippet, we changed the title to average target volume, the y-axis title to volume, and then in the data series, I'm calling the data series AVOL. And that will create the plot window. Next, we want to add data to that plot. And to add data to that plot, I have to calculate what the average volume of in my simulation domain is. And that's what I do in inside the step function in this code. I basically create a variable called AVOL. I measure how many cells I have, and the way I measure how many cells I have is by measuring the length of my cell list. And then I iterate over my cell list and add the target volume of each of my cells divided by the length of the cell list. And I added into this AVOL variable. And then I plot the AVOL variable. And again, just as a refresher from yesterday, if I want to add data points, I go to CC3D Python, scientific plots, add data point. And then I modify it such that the series, the name of the data series is the same that I specified in line 24. And that I'm plotting the right variable, which is in this case is the average volume that I call AVOL. Okay, I'm gonna end poll relaunch to make sure that everyone is with me. Okay, some people need a little bit of time to work on this. I'm gonna give it a couple of minutes. So just as a recap here, we've basically used the same simulation as before, as the previous exercise. We've just added a plot window in the start function to plot the average target volume. And we have calculated the target volume inside the step function, right? And I've plotted that, I added the data point to the plot that I created. So I'm gonna give people 1 or 2 minutes to catch up here. Also, as a reference, this code is available in the demo 3.1 3 or demo exercise 313. So now if I execute the simulation, what I see is that my average volume increases until my cells divide, increases until my cell divides, increases until my cell divides. And if it keeps going until it fills, until my cell fills up the whole simulation domain, and then it continues to increases, right? Because what I'm plotting is that target volume, and the target volume continues to increase. Now I'm gonna add a, what I'll do is I'll add an additional property to the cell dictionary that I'm gonna call pressure, and to add into that dictionary in the start function where I specify the constraints, I also define this entry or attribute in the cell dictionary that I'm calling pressure. I'm also creating an additional second window to plot the average pressure. And then I'm calculating in the step function, I'm basically reusing the code that I had already there, but to calculate the cell pressure instead of the average volume to calculate the average pressure. So in this part of the code, what I do is that I iterate over the cell list, and then I define the pressure on the cell dictionary to be this formula, which is a formula for pressure that we introduced earlier. And then I calculate and plot the average pressure of my cell. So this code is doing two things, right? It's first calculating what the pressure is that each individual cell is in. And secondly, it calculates what the average pressure in my simulation domain is. So what I'll do is I'm gonna open this exercise, this demo 313. And now we can see the full code there. So I'm defining this this entry in the cell dictionary called pressure. I'm creating a window to plot the average pressure. And then I am calculating both the average volume and the average pressure in my simulation domain, and I'm plotting them in their respective windows. Please do notice that when I create two different windows, I have to name them differently. So the first window, it's named self.plot_win. The second window is called self.plot_win2. And then accordingly, every data series and data point has to be referencing the right plot window that I want to add that data to. And then if I run that simulation, now I have a average pressure and average target volume. And well, in this particular version of the code, I already modify it. But if I don't modify it, what I should see is that the average pressure and the average volume, well, the average target volume will increase, and so will do the average pressure. So now I can modify the growth rate based on this property that I'm calling pressure. And possibly the simplest way of doing is to do it linearly from a reference pressure value. And the reason I'm using max is because I don't want my target volume to decrease. So if the pressure is over 70, which I chose basically by looking at these plots, then my cell target volume will be, the increase in my target volume will be 0. And that's the way I can control whether my cell whether the target volume continues to increase after this pressure is above some threshold, right? So in this case, the threshold is 70, and if the pressure is above 70, then the target volume stops increasing. And that's what I see if I run this simulation. So now the cells do fill the simulation domain, but the target volume stops increasing after they have filled up the simulation domain. And I'm gonna relaunch the poll just to make sure that everyone has been able at least to run this code, because I've uploaded it, and at least that you are that mark yes is you're being able to run the code, and at least you understand what the code is intending to do. Yes, I can definitely show the code again. So here is a part of the code where I modified the growth rate. Let me decrease this a little bit to make it a function of the pressure the salt is experiencing. So 70 was just a number I chose randomly. I chose that from the previous results. I saw that before the cells fill up the plate, they never reach 70 here as the pressure. So I wanted to keep the pressure. I set that threshold that way by looking, but I mean, that number could be way better or less informed depending on the motivation for it. But I just did it by looking at that plot. Okay, so I still have 10 minutes. There's a couple of stuff that I still want to cover. So the next, what I'll do is the next exercise, I'll walk you through it, and I'll show you the code, but Giuliano is gonna be talking about it more in detail later. So I'm just, instead of walking through building the whole simulation, I'll just explain the code and explain what it's doing, because it's important for what Giuliano is going to be teaching later today. And then we will move on to the basis of chemotaxis. So far, we've modified our cell growth using contact inhibition, right? That by somehow modeling contact inhibition using this pressure, cell pressure, or property that we've defined. There's other mechanisms that control cell growth, and that is not just contact inhibition. Or availability resources is also an important mechanism that controls cell growth, and this is something that becomes very obvious in tumor spheroids, where you have nutrients diffusing in from the boundaries or surface of the spheroid, And you can see that at the rim of the sphere, cells do proliferate and grow and proliferate. In the interior, cells die because they are because of lack of nutrients, and there is like intermediate regions where cells are in quiescent cell without where they do not grow, but they where their cell cycle is arrested. And this is controlled by the diffusion of the nutrients from the exterior or from the surface of the spheroid to the inside. And we can model that in Compucell by coupling cell growth with uptake. And this builds on the ideas that we were discussing yesterday, where we created a chemical field and we had our cells uptake from that field. Now we can tie that uptake to cell growth and control cell division that way. And the idea is simple, and actually the code to implement that is not that complicated. All we need to do is to modify our cell growth by the amount that the cell is taking from the environment. So if the cell takes nothing, then the cell does not grow. In the simplest case, the cell doesn't grow. If the cell is taking as much as it can, then the cell grows as fast as it can. Additionally, there's nothing that prevents us from combining different control mechanisms for cell growth. So we could potentially combine pressure-dependent growth with what I'm calling here diffusion-limited growth. The syntax is quite easily. We're gonna use this uptake inside cell function that we saw yesterday, but we will also add this total count that allows us to recover how much and reference how much the cell is uptaking from the environment. And again, the idea in this code is that I'm going to walk you through it. We're not going to build it together, but we create a simulation domain just as in previous exercises, and then we add a nutrient chemical field, and then we've had our medium secrete this field at a constant concentration that we set to be 1. And again, for those of you who were not able to join us yesterday, you can there's videos of those presentations where we discuss how to create chemical fields and how to set the secretion options. And then what we will do next is that we take from that field, and then we record how much we're taking from that field, and we create an additional visualization field that allows us to see visually how much cells are taking from the environment. And finally, we modified the growth rate by how much cells are taking. So to see this code, we can come to, we can open the demo 314. And in the XML, for those of you who were with us yesterday, you will see it's just the same as the previous simulations, except that now we have added this chemical field by specifying the diffusion solver. I'm sorry, my computer. Do you guys still hear me?
Yes.
Okay. I think it's my nano hub. My nano hub is acting up. So I'm gonna have to terminate it and run it back. But coming back to this, we've just added a chemical field, and I'm not gonna go into the details of this because this was discussed yesterday. Oh, it's my email. And then with just 1 second, let me make sure that my internet is working. Okay, do you hear me now? Good. Okay, I'm waiting on NanoHub to load again. But the idea in this code is that we've created a I'm sorry, I'm relaunching the end of model questionnaire. So we are creating a chemical field, and we're having our medium secrete. And then we've also enabled the plugin stepable, the secretion plugin in the XML. And in the step function of in one of the step functions, we have make our cells take up from their environment, but and ran the simulation. And in this point, what you will see is that the cells grow, but they take up from the environment. But so far, I haven't tied up how much they take from the environment with how much they grow. So basically, they create this gradient from the edge of the blob to the interior, because they are taking up some of the nutrients. And then next, what I do is that I create an additional cell dictionary entry called Uptake, because I want to record how much my cells are taking from the environment. And then instead of using the traditional uptake function, I'm gonna use now this uptake inside cell total count option, which allows me to retrieve the number or how much the cell is taking from the field. And I'm passing that information to the dictionary entry. And I'm creating an additional field that would allow me to see how much my cells are visually, how much my cells are taking up. And I do it by going to CC3D Python, extra fields, automatic tracking, track scalar cell attribute. And notice that this has to be in the init function. And then I call that the field name uptake and the attribute name, which has to correspond to the attribute that I've created in the cell. I call it uptake. And when I ran that simulation, I can now display my cell field, my nutrient field that I see that it's decaying as the blob grows bigger. And that can I also visualize how much my cells are taking up from the environment. And I can see that the cells at the edge of the blob are taking more than the cells at the center of the blob. And now I can finally modify how the cell growth by how much my cells are taking from the environment. And I do it, this is the same equation we saw earlier. I do it by dividing by this, how much my cell is taking up by a reference number. Why the nutrient levels in the corner remain high? Because the setup the setup for this particular simulation is that the medium is the one secreting, right? So at the edge of at the edge, the boundary of the cell blob is with medium. Medium is secreting, so medium is always high, especially because I set it at a constant concentration. But as soon as the chemicals start diffusing from the medium, from the interface here inside the blob, then it starts being consumed by cells. So now I can modify how much the cell growth based on how much it's taking up. And I can play with how the diffusion coefficient affects how much my cell is growing. So if I make my diffusion coefficient smaller, that means that the molecule or the chemical field diffuses shorter into the blob. And I can see here that only the cells at the edge of the blob are taking up. So those are the only cells that are growing. And then as opposed to when my diffusion is bigger, that my chemical is able to diffuse further into the blob. And so cells are able to grow, cells deeper into the blob are able to grow as well. So I still have the chemotaxis to introduce, but what I'll do is I'll stop here, and Gilberto is still gonna talk about how to implement chemotaxis later today.
Josh, I think
Yep.
chemotaxis is important enough that I would take the beginning of the next module and use it there.
Okay, I can certainly explain it in the next
So
module.
during the break, work with Giuliano to put that in at the beginning.
Okay, absolutely. And but before finishing here, I just want to make sure that I explain this well. So remember what I did is I have the chemical field being produced in the medium, and then the cells take up from that field. How much the chemical diffuses into the blob controls which of my cells grow and at what rate. If it diffuses over short distances, then only the cells at the edge of my blob are able to grow. If it diffuses deeper into the blob, then more and more cells are able to grow and divide. And I'm gonna stop here, and then I'll continue with chemotaxis on the next module. So James, I'm handing back to you.
Okay, thank you, everybody. Please fill out the little questionnaire on this module, if you would. And we will start back at 12. It's 12 o'clock, so we'll start back at 12:15 for the next module. Thanks very much to everybody.
Hello, everyone. We're about to start in 1 or 2 more minutes to give everyone time to sign back. Okay, hello, welcome back, everyone. This is the module 3.2. It's gonna be part 1. We're continuing with some of the discussion from the previous module. In this particular part, we'll introduce the idea of chemotaxis, and we can control certain properties and behavior based on movement due to chemical gradients. I'll try to be short in this part because this is an idea that Gilberto will come back over later today. But I still wanted to have some time to introduce the basics before Gilberto introduces a more complicated model. Just as a reminder, the presentation is being recorded and live streamed. As I mentioned, this is module 3.2 is a avascular tumor simulation, but we're running over some of the stuff that we left behind in the previous module. I'll try to be short and hand it back to Juliano after introducing chemotaxis. So what I'll do in this part of the module is I'll introduce the idea of cell movement due to chemical gradients, and I will show how this can be implemented in Compartmental on a linear gradient, and how we can construct a cool and very simple simulation of bacterial movement in a nutrient that is fixed to, let's say, extracellular matrix. So biological cells alter some of the behaviors that cells alter due to or in response to signals and gradients. We saw earlier that cells can do this, can control cell division and proliferation due to changes in signals. Another type of behaviors that cells can change is movement. We will, and there are different signals that cells can react to. They can react to how rigid their environment is, and we call this deutotaxis. They can react to electrical fields. They can react to changes in temperature, moving, let's say, away from areas that are too hot or too cold. And they can also move in areas that are high or low in, let's say, light sources. But in general, since we have introduced the idea of the chemical field, we can readily implement in Compartmental cell movement resulting from an exterior factor due to the gradients of some chemical field, and we'll call this chemotaxis. Chemotaxis is when is the movement of cells from due to exposure of chemical gradients, and these are ubiquitous in biology. A classical example is the response of discotilium to sources of CAMP. And this individual movement of cells due to the gradients lead to very interesting large-scale behaviors. And I posted a couple of links in the slide deck that show examples of cells chemotaxing due to some signals that have been added to the medium in which they live or move. So to model chemotaxis, all we need to take into consideration, or all we need to think about, is the fact that cells will move in response to how the gradient of the field is, or the chemical they are in is. Such that if, for example, the gradient is steeper, right, or the concentration is changing faster, then we will expect our cells to move faster up that chemical gradient. If the gradient is very shallow, then it's harder for the cell to detect that gradient, and so the cells will move a little bit slower up that gradient. In Compucell, we modeled that by adding an additional term to our energy terms, what we call the the chemotaxis constraint, and we control that chemotaxing by adding this lambda chemo that controls how sensitive the cell is to the gradient. So there's two parts to the force exerted over cells due to the gradient. One is the steepness of the gradient. If the cell is if the gradient is very shallow, cells don't move that fast, as opposed to if it's very steep. But also the sensitivity that we control through the lambda chemotaxis. The cell can be in a steep gradient, but it's not. If it's not sensitive to that gradient, then it won't move up to the gradient. But if the cells is very sensitive, then it will move up to the gradient. So it's a combination of the sensitivity and the steepness of the gradient that it will induce the cell movement. Right, this is the same idea. We call this force, or this energy term, the chemotaxis effective energy. Notice that if the lambda chemo is negative, if it's negative, what our cell will do is move from areas of high concentration to areas of low concentration, in which case the field is acting as a chemorepulsant. If the lambda chemotaxis is positive, then our cell will move up the gradient, in which case our chemical field is a chemoattractant. There's multiple ways in we can implement chemotaxis in Compucell. For the purpose of this model, we will do the simplest type of chemotaxis implementation, which is done in the XML specification. This is sort of like the code that we will have to fill up. We'll have to specify what the name of the field is that will cause our cells to move, what the type of the cell is that it will be moving, and then the lambda chemotaxis, as described in the previous slides. So for this exercise, what we will do is that we will create a we will create a simulation domain, and then we will add a chemical gradient that decrease with a linear decrease, and this is based on based on the presentations from yesterday, again, for those of you who weren't able to be with us yesterday, I would direct you to the modules, the discussion on this, the modules yesterday, where we showed how to implement where we can implement a linear gradient based on the properties of the field at the boundaries. Then what we will do is, after creating a linear gradient and initializing a single cell, we will introduce the Chemotaxis plugin, and we specify the lambda chemotaxis. And then we will plot the position of the center of mass of our cell as of in the X domain, and we'll run the simulation for different values of the lambda chemotaxis. For these first parts, first part 1 and 2, I'm not gonna go into detail because, again, this is something that was built up on from the presentation yesterday. But I've already produced the code. If you wanna produce it by yourself from scratch, all you need to do is to run your simulation, to create a new project. Let's call it Chemotaxis. I'm sorry, I'm not sharing my full screen, I'm sorry. So I'm creating a new simulation. And I'm calling it Chemotaxis. I'm sorry, I have to choose the right directory to create it. And then I said that we are gonna set a 200 by a 200 lattice. I'm gonna create a no flux in this case, but it doesn't matter at this point because we're gonna stop the simulation before our cell reaches the edges. I'm gonna use the blob initializer to start our cells, to initialize our cells. I'm gonna create a single cell type called cell. Again, pixel copy of 3, I keep forgetting about it. But yeah, it's always preferable to use a pixel copy range of 3. I use a cell type called cell. Again, I have to create a field, let's call it here nutrient. And use the diffusion solver FE. And then here I will select contact, Chemotaxis, volume flex, center of mass. I need to make sure that I have it enabled so I can actually plot the center of mass of my cell. When I've selected Chemotaxis, when I hit next, instead of ending the wizard, I'm gonna have an option to specify what the Chemotaxis plugin form it will be. And I said that we will have let's start with a lambda of 0 because we don't have our cells to move, and we'll modify directly into our XML. And the cell type we want is our cell type cell to move up the nutrient field. And we're just gonna use regular Chemotaxis for this example. And then once I do that, I can finish my simulation. And this is the same as exercise 3 0.15. Okay. So when I load my XML, I can see now that in the XML, and this is based on yesterday, that's why I'm not discussing it in detail today, but I'm creating, I am setting the conditions such that I have a linear gradient starting at one edge and ending on the other edge. Yes, the chemotaxis towards means when cells are in contact with, you can control what happens to cells when they are in contact with other cells in terms of the chemotaxis. So if I specify that the chemotax towards medium, that means that once cells has reached other cell types, they might stop chemotaxing or moving. So I have in chemotaxis towards, I have to specify which cells once my cell is in contact with cells, which cells inhibit their chemotaxis and which cells do not. Since in this particular case, we just have one cell and it's surrounded by medium, I didn't care to specify that, but that's a little bit more complicated. For more complicated simulations. So again, I'm not gonna go into the details of the chemical field specification because that's discussed yesterday, but those specifications will guarantee that you start your simulation with a linear gradient and with that cell at the edge of your simulation domain. And now in that XML, you can see that we have the chemotaxis plugin. And if you started with lambda 0, you should see it here. If I saw in the comment, someone says that it's not loaded into the XML, as you can always come back to CC3DML plugin and hit on chemotaxis, and then that will load the chemotaxis plugin for you. If you forgot to specify, let's say, wizard. So far, what I've done is some parts based on yesterday, I created a linear gradient and I've added a single cell at the edge of the simulation domain. And now I specified that my cell has a lambda of 500. So how do I specify what field is responding to? It's called field here. It's just, I'm sorry, in the example that I showed, I call it nutrient, but in this case, I call it field. That was from yesterday. But I specify what field the cell responds to in this part of the plugin. So this says that my cell type cell is responding to the field, unfortunate choice of name, with a lambda chemotaxis of 500. And that's what's explained in this simulation. Now, if I run that simulation, what I'll see is myself moving from one edge of the simulation domain to the other edge of the simulation domain. Yes, I can certainly show the XML. Remember that the XML has that part where I'm creating the linear gradient, and I'm actually not going into the details of that. That's why I wanted to share the base code with you. But what is important for the purpose of this module is that we specify the Chemotaxis plugin. Just make sure that the name of the field here and here matches the name of the field the cell is responding to inside the Chemotaxis plugin. So when I start the simulation, I can see that my cell starts. What should I put in chemotax towards? You can put medium, or you can leave it blank. I do not have it in my specification. But as I said, you can put medium because it means that it's gonna move through the medium to get up the gradient field, the gradient of the field. So now I can show my linear gradient, and I can see my cell. And if I execute the simulation, I can see myself moving up that gradient. Right. James is right. That is the right definition is of the chemotax towards. It specifies the surfaces on which the cell is going to attempt to move towards. So if I specify medium, it means that it will try to move over medium. If I want to be able to chemotax over other cells as well, then I will have to specify what cell types my cell is able to move. Right, that's exactly what James is specifying there. So in this particular case, I can see that my cell is moving up the gradient, but it's moving very slow. If I increase the chemotaxis, the lambda chemotaxis, which I can do here in the model editor, then I should see my cell moving faster up that gradient. So now I can see my cell actually moving much faster up the gradient. And if I set it to 5,000, now I can see my cell moving much faster up the gradient. And I'm gonna stop my simulation there. Yes, that's what I'm doing. To see the cell on top of the gradient, you select the field in your visualization window, but just make sure that your cell borders are checked up. If your cell borders are unchecked, you won't be able to see the cell on top of the gradient. Okay, now in the next part, what I'll do is I wanna plot what the center, I wanna quantify the movement of my cell. So I'm gonna plot the center of mass of my cell as a function of the position. And again, this is a refresher. I create a plot window in the start function of my steppable. And then what I do is I create a data series called POS. And then in the step function, I iterate over my cell list and I plot the time and the position of the center of mass on the X direction. And I plotted just on the X direction because that is the direction of the gradient. If on the Y-axis, the cell might move up and down, but that's just gonna be due to random cell membrane fluctuations and not due to the chemotaxis. So I just plot the X-axis to see the movement due to the chemotaxis. And now if I execute my full code with the plot, what I should see is that now I can see how the center of mass of my cell is changing due to the chemotaxis, and it's decreasing, right, because it's moving left. And you can see that there are some fluctuations up and down here. Anyone has a guess as to why that is? Any guess why are we seeing this wiggle in the position? So what's happening here is that, right, because the cell movement is right, because the cell movement is composed of two components, right? I have the cell movement due to random fluctuations in the membrane, and I have the cell movement due to chemotaxis. And if the movement due to chemotaxis is relatively small, then what I should see in my movement is both the combination of the random fluctuations in the membrane plus the chemotaxis. If I were to increase the chemotaxis, the lambda chemotaxis, I'm sorry, I stopped it, but I shouldn't have. Okay, I didn't stop it. So if I were to increase the lambda chemotaxis, let's do it to 3,000. Then the cell is gonna move much faster, right? Because the velocity of the cell is the slope of this curve. And now there's less wiggle in that movement, and that is due to the fact that the contribution of the movement due to chemotaxis is much bigger now compared to the random fluctuations. And you can see that the velocity changed when I changed my chemotaxis. The velocity here was about, let's say, 20 pixels per 100 Monte Carlo step. Here it was about 100 pixels over 100 Monte Carlo steps. Okay. So I'm gonna leave this to you to play so you can restart the simulation with different values of the lambda chemotaxis. You can modify them either on the model editor, or you can come here to the plugin specification to change it. Yes, so the chemotaxis is indifferent as to where the field is coming from, or even if the field has a particular shape. In this particular case, we set the shape to be linear, but the cell can react to any type of shape of the chemical gradient or what the source of that chemical gradient is. So I'm gonna move on to the next exercise because this I'm gonna treat it as an example so I can hand back to Giuliano for the avascular tumor. But in this case, what we will do is that we will construct we will build a simulation that it resembles bacterial movement with a nutrient that is bound to, let's say, an extracellular matrix. And the way we will do that is that we will have, we will initialize a simulation with 100 by 100 lattice and periodic boundary conditions for the cells. We will initialize a single cell type, amoeba or bacteria, and a single field nutrient. And we will have contact, chemotaxis, and volume constraints. Then in the XML, for the field, we would set the global and decay coefficients to be 0. So that will mean that we will have whatever field is there won't diffuse and won't decay. And this is how we model this matrix-bound nutrient. But we will specify different diffusion coefficient and decay coefficient inside the cell. And this, again, this takes us back to yesterday, where we were specifying diffusion coefficients and decay coefficients by cell type. We will set the field boundary conditions to be periodic, and we will initialize the concentration to be 1 everywhere. So this is with these initial conditions and diffusion and decay coefficients, what we should see is that our full simulation domain has an initial concentration of 1, and then we will place one cell in the center of that simulation domain and let it move due to the chemical gradient. But the chemical gradient will be created by the cell itself, the cell itself as it moves through the simulation domain, because otherwise the chemical field does not diffuse or does not decay. So what I'll do is I'll walk you through that code. This is exercise 3.316. So in the XML, we have created our simulation with our simulation domain as specified in the instructions. We have initialized our cell, and we have specified our volume constraint here in the XML because we're not changing it, so we can specify it here. We have the constant energies, and then we have specified a chemical field called morphogen that has a global diffusion constant of 0, a global decay constant of 0. So that's how we model the nutrient-bound, matrix-bound nutrient, but it actually decays inside the cell. It diffuses and decays inside the cell. And we've set up the field to have an initial concentration of 1, and then we have specified that our cell can move due to can chemotax in this gradient with a lambda chemotaxis of 200. And there is really nothing else that we need to specify this simulation. It's very simple. It's complicated conceptually, but simple to implement, and actually produces a very interesting simulation. Let's run it here. So we start with a cell in the center of the simulation domain. And as I said, there is field, the quantity of the field is 1 everywhere. And I started the cell, and once I start the simulation, the cell starts consuming from that field as it moves and chemotax in the field. Remember, the reason it's not diffusing inwards here is because it's membrane bound, and I set that by setting the diffusion coefficient to be 0. But inside the cell, the chemical field can actually diffuse and decay, and that's why the cell can construct its own gradient. And you can see that it's very interesting that the cell has both types of like persistent movement and then some type of tumbling and moving around. So it persists on one direction, it tumbles a little bit, and that tumbling is due to random membrane fluctuations, and it continues moving. But you can also see that the cell avoids region it has already been to, because it already consumed the available nutrients in that region. Okay, and what I'll do is I'll stop here. I've been talking for half an hour now, and I want to give Giuliano time to present his avascular tumor demo, but I'll be happy to go over this particular simulation. I know some people are interested in this type of simulation in the hackathon, and this is a nice example of how can be implemented rather easily. Yes, it is because the cell eats up. In this particular case, we implemented it by setting some decay inside the cell, but we could also do it by setting it to be some uptake that we didn't do, just to keep the simulation simple. But yes, when I say creates its own gradient, it's because the cell is taking up or eating up the nutrient. I'll stop here and I'll hand back to Juliana now.
Hello again, everyone. So we'll try to get a avascular tumor demo done using the concepts of diffusion that we were talking about. I'm sorry if I go a bit fast today, but I have less time than I expected. So,
Juliano, it's okay. If you need more time, I would make sure everybody gets through it. And if we move some material from this afternoon to tomorrow, I think it's better to take the time we need so everybody follows.
okay. Okay. Okay, so most of the lecture is, well, going to be exercises building up the simulation of a vascular tumor. We are going to have 3 cell types. We are going to have cell division. We are going to have cells uptaking nutrient from a chemical field. We are going to see how to kill cells and how to mutate cells, hopefully. So in a avascular tumor, we see 3 layers of cell behaviors, with proliferating cells outside where they have enough oxygen and nutrient, then quiescent cells where there isn't so much, and finally necrotic cells when, well, they don't have enough to live. And as we have the diffusion parameters, we can set those so that we know where, more or less, inside the tumor, the chemical is going to be at a certain level. So in this image here, we can see, I don't know, 4, 5 cells in that are still proliferating, at least according to Garcia and all. And we're going to use that to define our diffusion constants of D and gamma. And we're going to define those in the natural units of pixels and MCS. Another thing that we'll do is, instead of having the nutrients come from the very edges of the simulation, we are going to have the whole medium be the supplier of nutrient. That way, we're going to see the tumor cell self-limit itself. And I think the resulting movies are quite cool. So actually, let's get properly started. So I already have Threaded open here on NanoHub. And as usual, we are going to start a new project. I'm going to select where I want to save it. I'm going to create a new folder here. 3, 2. Oh, I need to actually create the folder. There we go. And I'm going to call my simulation avasc tumor for short. I have to drag these things to make them show properly. We are going to leave the dimensions, the default of 256, but we are going to use periodic X and Y. And Josh says that it's best to have a pixel copy range of 3. I personally prefer 2. So a bit of a difference. And instead of having the rectangular slab, we are going to use the blob initializer. After all, our tumor is roundish, so it makes sense to start with something that is round. We have 3 cell types. The proliferating cells, which I'm gonna call prolif for short, the quiescent cells, and I'm again shortening, and the necrotic cells. And we are gonna see how to change cell types in between those based on the chemical field that they are exposed to. In reality, as this figure implies, we have both waste and nutrients in different gradients. And not only nutrients, but we also have oxygen, and not only waste, but we have several types of waste. But to keep things simple, we are gonna govern the behavior with only one field for the nutrient. And we can see that that's enough to get the behaviors we want. And again, we can always complicate things after we get something working. So only one chemical field, nutrient, and I'm using the default solver. As for plugins, we're gonna use contact as usual. We are gonna use secretion for when we do the uptake. Uptake is just negative secretion. And we also have cell division, so we select mitosis. And now we only have the option of volume local flex. We cannot use the volume non-local flex. And that's the final plugin we're gonna use. So contact, secretion, mitosis, volume local flex, and center of mass, but that's selected by default. And now we come to secretion. So I've mentioned that we're gonna have medium be our source. And this allows us to set the XML properly for that easily enough. So at least for me, I already have nutrient selected and I already have medium selected there. And what I'm going to say is that it's going to be set to 100 for 100% of nutrient. And I'm selecting constant concentration there. So I'm imagining that the influx of nutrients is enough always to keep it level. Now we actually have to click add entry here and it shows up. So again, I already had nutrient and medium selected. I set 100 there and I selected constant concentration and add entry. And if you made a mistake, you can clear table here. And that's it. So from the start, well, I gave it a name and a location. I left the dimensions default, periodic, X and Y, copy range of 2, but shouldn't change much by using 3. 3 cell types, prolif, quiesce, and necro. 1 field of nutrient. Contact, secretion, mitosis, volume local, and center of mass. Nutrient secretion of 100 by medium, and that's set here, selecting the field, selecting medium, 100, constant concentration, add entry. And that's it for configuration. If we told Compartmental to run this, it would run, but to edit by default pastes a bunch of stuff that we are not actually going to use, like here on the nutrient field. It has all the code to have different cells with different diffusion coefficients. But for now, we're gonna keep everything the same. So I'm just going to delete those tags. And there's, oh yeah, so after deleting those tags, we come to our first decision for the diffusion constant and decay constant. So I like my cells to be 64 pixels, but that's me. And going by that image that I showed earlier, we have about 4 cells deep until they transition. So I'm gonna set things up such that our diffusion length is about 4 cells. So if cells being 64 pixels and roundish, their diameter is 8 pixels, not really, but It's fine. And so we'd want our diffusion length to be 32 pixels. And since the diffusion constant and the decay constant are related with, well, the diffusion length is the square root of the diffusion constant divided by the decay constant. So I just rearranged things here and I already squared the diffusion length. We want a diffusion constant that is about, so let me type it here maybe. So the diffusion length is the square root of d over gamma. So if I pass the square root to the other side and gamma to the other side, I have that d is dl squared times gamma. And we know that dl should be 32 pixels, and 32 squared is 1064. Gamma is the decay constant. And so, but 1024 is about 1000. So we want our diffusion constant to be 1000 times greater than the decay, which is close to the defaults here. And I could just set this to e to the minus 4 and we'd be good to go. But in that case, the diffusion would be a bit slow. So as the tumor consumes nutrient, it would take longer for nutrient to come in, and that could have unintended consequences. And well, I've set the parameter I have in the presentation using 10 and 0.01. You could use 1 and 0.04 or things like that. But then the thresholds that you need to use to control the cell behavior would be different. Next thing the next thing I already did, which was deleting all of the diffusion coefficients for the cells individually. We are only having the global one. And we are going to start our simulation with, we are going to use the initial concentration expression. And we are going to start with 100 units of nutrient everywhere. So I'm going to uncomment this line and replace that x times y by 100. Okay. And we can also see here that inside the diffusion solver, there is some secretion data, but there's also the secretion plugin here, kind of redundant. And well, cells should consume nutrient. They shouldn't secrete nutrient, right? So I'm just going to comment these lines out. And I'm pretty sure that we do need the secretion data tags inside the diffusion solver for things to work properly. So again, I select those lines and add it and block comment, since commenting on XML is cumbersome. And since we have medium secreting the nutrient, medium is our boundary for the chemical field. And so what we are going to use here for boundary conditions for the chemicals is periodic in both X and Y. So again, I'm going to comment that lines that set constant value or constant derivative for X and Y. And I'm going to uncomment the periodic tag there and here. Okay, so again, everything that I did in the diffusion plugin. First, I set my diffusion constant to be 10 and my decay constant to be 0.01. I've set the initial concentration to be 100 everywhere. I've removed the extra diffusion and decay constants for each cell type. And I remove the secretion of nutrient by cells, which doesn't make sense. And I've changed the boundary conditions to be periodic in X and Y. And one final change that we are going to do here in the XML is to the blob initializer, which is first to have the cells begin with 8 pixels, because that's what I like. And instead of starting the simulation with randomly placed per leaf, true, I meant 1 extra 0. These should be 1,000 times gamma, is that right? Yep, yeah, no, you're right. Yep, it should be 2, not 3 here. Thank you. You should have just typed over, copied from my slide. Trust my slides, not me. But yeah, and then in the blob initializer, we have all cell types here, but we are going to start with all tumor cells being proliferating, give the tumor a chance. But one small parenthesis that I could make here is that if you want, so if I have prolif and quiesce here once each one, I'm going to have half the cells be proliferating and half be quiescent. But if I have proliferating twice, in this case, 2/3 of the cells would be proliferating and 1/3 would be quiescent. And in this case would be 3/4, 4/5, etc. But I want 100% to be proliferating. And if now we go to the steppables, because we selected mitosis, we can see that the steppable structure is a bit different. There are 3 steppables, one for the constraints, one for growth, which is already active even though we didn't select it, and the mitosis. So first, we don't want our cells to be 25 pixels, we want them to be 64. We could leave lambda to be 2, but I prefer 8. And I know that the simulation will work with 8, but I don't really see a reason why it wouldn't work with 2. And here in the growth, we're going to leave as is. We're going to let things grow for now. But here in mitosis, we already have a problem because our cells are starting with 64 pixels. So if I just show it to you, what would happen here? Start with our cells all pretty and nice there. And if I step it once, they all immediately divide because the condition for division was assuming a target volume of 25 instead of 64. So instead of 50, 2 times 64. And we can see that the other thing that happened here was that half of the cells changed type after division. And we don't want that behavior, at least to start, for this simulation. And that behavior happens here. So I'm just going to, again, comment these lines out. And now if I do it again, and we go. Okay, things look all right. My cells are there and they're still growing because I left growth alone. And one thing that I'm going to do, and we already saw some division there, and I'm gonna have a second graphics here for the chemical field. So we can already see how the tumor is gonna self-limit because we have 100 everywhere in medium and it's going to stay that way. But inside the tumor, things are actually going down. Yeah, and here's what we are seeing here. Okay, so we need cells to change their type based on them having enough nutrient available. So we want one transition from proliferating to quiescent, one transition from quiescent back to proliferating, and another transition from, well, from quiescent to necrotic, but really should be from anything to necrotic, but quiescent to necrotic will work in our case. And we also want the proliferating quiescent and quiescent proliferating transitions to be not at the same level, because if you imagine that if the concentration is at that level, random fluctuations can make cells flip-flop between the two behaviors. So we're gonna set the quiescent to proliferating to be a bit higher than the other one. So it takes a bit more effort to begin proliferating. So we're gonna do this in the growth steppable, since we already have a step function here. And we are going to replace this loop over everything with two loops, one over proliferating and one over quiescent. So I'm gonna comment out this loop here. And I'm going to add my two loops. So first, asking to edit how to visit cells of a given type. And again, it pastes this. We could do both types at the same time, but it's best to do one at a time for this simulation. It doesn't matter which stepable we put it in, as long as it's inside the step function of the stepable. So in this first loop, I'm gonna go over the proliferating cells. And instead of asking to edit again, I'm just going to copy the loop and just change this to quiesce for quiescent cells. And yeah, to edit, and so now we actually need to measure the field and do something with that measurement. Thankfully, if we look down here, to edit already pasted what we need. So it pasted this command that grabs the field, all the values of it, and this command that actually gets the field at the center of mass of the cell. So we can just copy from here. First, I'm gonna copy the field definition to be before our loops. After all, we are gonna use this object field here in the loops. I just need to change the chemical field name to be nutrient. In this case, so one inconsistent thing is that cell types need to be all caps always, but fields need to be as you type them here. So here I have nutrient all lowercase NUTR actually. So in here I have NUTR as well, lowercase. And then to get the concentration at center of mass, I'm gonna copy this line here. And I'm going to paste it inside both of my loops. So one thing that I did here was I was printing the chemical concentration. But one thing that I forgot is that in NanoHub, unfortunately, we can't see the prints. If you are doing it locally, you could just print this. So what we are going to do is instead is remove those prints by commenting them out. And then we are gonna add a cell dictionary for that value there. So cell dict conc for concentration. And that should be equal to concentration at COM for center of mass. And I'm going to copy that here too. And now we can actually visualize those concentrations by adding a extra field. And extra fields can be added in any init of any steppable, and Compucell will figure things out. So I'm coming here into the init of the growth steppable, but I could do it here as well, as long as it is in an init function. And I'm gonna ask to edit how to do extra fields, automatic tracking. We have these old commands for backwards compatibility, but they are cumbersome. And this is very not cumbersome. So I just click track scalar attribute. And what this command does is that it's looking for this attribute name inside the cell dictionaries and is going to display it in player. So the attribute you need to look for, in my case, is conc. And I'm gonna also name my field conc. And now if I run, I have both nutrient here, but I could also add another window and have conc. Now I'm gonna hit dial. I'm gonna go over everything again. So now we can visualize the concentration that they're sensing at their center of mass. So what I did in Python was, first, I removed, where was it? Here. I removed, conc is just the concentration that I measured of the nutrient field. They look similar, but because I'm measuring the concentration at the center of mass, only we can see that the jumps are discrete, whereas in the actual chemical field, it's a slow gradient. There you go. So in the steppables, what I did was I changed the constraints in the constraint initializer to be 64 and 8. I then changed the trigger for cell division to be 128 pixels instead of 50. And I removed the change of type with cell division. And then I removed the loop in the growth step over all cells and added 2 loops, one for the proliferating cells and one for the quiescent cells. And then I noticed that there was already some code here that I actually could use. And I copied this, well, first I copied this declaration of the field values here. So I have the field values of the nutrient field in the handle field. And I also copied over the command to measure at the center of mass of the cell. And I'm passing that measurement to this dictionary here. And then I came back to the init function and I asked to edit how to track a custom field automatically here. And as you can see, you could also do vector cell attributes. So for instance, for bias yesterday, we could have an extra field that actually displays the bias vector. And exercise pre-launch. Yes, the code is available at the demos files. Yes, good question. It's here. And it is how the code is going to look at the end. So you can use that to cheat a little bit if you want or need to. No shame in that. Let me actually just make your life easier. Okay. So next thing is actually to define, well, the thresholds of each of the transitions. I did it here in the growth table, but I actually don't want to do that now. I want to do it in the constraint initializer, since we have it. And this way I can show you how to use the steppable shared dictionary thingy, which is very useful. So first things first, I'm gonna define the proliferating quiescent transition to be a fraction of 100, the threshold. And the threshold that I selected is dividing by the half of E. And that's just a property of the diffusion length. And so A diffusion length, the concentration is gonna fall by a product of E, but I'm adjusting it a little bit because I know that works better. So here I'm using numpy E, so I actually need to in the start. Yeah, in the start, I think it wouldn't work in the init, but I'm not sure. So I'm gonna import numpy as np. So I'm gonna have 100 divided by half of E. And I actually need to remember to multiply my numbers. And then I'm defining the QP transition to be just a bit higher than the other one. So an easy way is to just multiply PQ by a bit more than 1. And finally, the transition to necrotic, which is QN, is going to be 100 divided by E full E. And now we actually need to save all of these numbers into something that we can access in the growth steppable. And again, Twedit knows how. CC3 Python, this one I don't remember exactly where it is, but I think it's in simulation. Yes. So CC3 Python simulation, shared steppable dictionary. So the shared steppable vars is the dictionary that all steppables share and know about and coordinate. So in here, I'm gonna add a key for each of them, which I'm just going to use the same variable name, and set that to be PQ. And same for the other ones. If someone is curious how I multiplied my line that fast, it's Control D. It copies the line that you are in below it. So again, I imported numpy. I defined my numbers. I asked to edit, what was the name of the dictionary that all steppables know again? Which is in CC3 Python simulation shared dictionary. and just giving them keys and the numbers. So now we actually have everything we need to have our cells make decisions about their life. So if we go back here to the growth steppable, in our loop over proliferating cells, we're going to make them quiescent by adding a if statement. Oh, it's not there? The dictionary exists. It's just that Reddit doesn't know how in that case. Let's see if I can copy this outside of. Well. That's the name of the dictionary. So here in the loop over proliferating cells, I'm gonna tell them that if the value here in this dictionary with key conc is less than the number here in this shared steppable vars, then it should change its type. And type is a cell property that is easy to remember. But again, if you don't, and I surely don't sometimes, it's Python cell attributes cell type. So now then we know that cell type is accessed by cell.type. So we actually don't care what the value of cell type is right now. What we care is that to change it from whatever it is, which is prolif, to quiesce. Now, just to test, I'm gonna run this and we are gonna see some cells become quiescent eventually. Yep, there we go. And I'm going to do a similar thing with the quiescent loop, but in this case, it would be another threshold, but I can just copy over this if and just change the key here from PQ to, well, first let's do the reverse, so QP. And in this case, we're going to change the cell type from whatever it is to prolif. And now we need a second if in case this isn't true. So I'm gonna add a elif, well, not capital, elif. I'm just going to, well, one thing that I was going to make as a mistake here, I kept the less than. I shouldn't have kept the less than. There should be a greater than, since it's going back into high levels of concentration, right? But now I can safely copy this one with the less than, since this if is going to turn the cells necrotic. So this transition is QN. Then cell type is going to equal self dot necro. Okay, so from the top, what I did was I came back to the constraint initializer. I defined my threshold numbers here. I asked Reddit how to access the dictionary, but it's also in chat now, so you can just copy from there. And it, copied those numbers into different keys of this dictionary. And then down here on the loops, I'm using the numbers associated with those keys to change cell type. So in the case of the proliferating cells, if the concentration they are sensing is below this number, they become quiescent. In the case of the quiescent cells, if the concentration that they are sensing is above this number, they become proliferating again. And if the concentration that they are sensing is below this other number, they become necrotic. Now, if I run this again, we already are going to see the 3 layers forming. Yeah, another window here for the conch and tile. Sure. Just, yeah. So people that done it, you can let it run and you see the 3 layers already. Actually, let me make this a bit bigger.
Could you define the shared dictionary in the init functional other steppables?
Yep.
So was there any specific reason that you put at the first one? I mean, the Python starts from reading from the top, you mean?
Yeah, I've put it here because this steppable is named constraint initializer. So I thought it would make sense to initialize a constraint here.
Okay.
But we could have just done it all here in growth steppable. And yes, we could make the transition between cell types stochastic. I would still make it stochastic dependent at least a little bit on concentration, but we could have, instead of having a one threshold for all the cells, we could have it defined on a cell basis and have, instead of having the same number, varied a bit randomly. And actually that's a exercise later when we add mutation. So if I just come back here, I still don't have the 3 layers. That's fine. Yep. Yes, I'm gonna upload what I write here in the class later there, but it looks a bit different, but not really. The only difference is that things have different handles, but it's exactly the same code. So now, well, we removed growth, but we can add that back. So we want the proliferating cells to grow. So we could naively just, add it here before the if, but that would mean that some cells that turned quiescent would also grow. That's not really what we want. So what I'm going to do is add a else statement to this if here for the proliferating cells. So now what the proliferating cells will do is that if the concentration is below the threshold, they become quiescent. And if it's not below that concentration, they are gonna increase their target volume, which we can remember how to access by asking to edit for the cell attribute target volume. And we are going to increase it by, say, a half every time step. And we also want to do something similar here inside the quiescent cell loops because, well, some of them just became proliferating. So inside of that, if that changes the time to prolif, I'm just copying over the growth in target volume. So now if I let this run again, And I don't know why CompuCell keeps forgetting that I want 3 cell fields, not 2. We can see that the proliferating cells are getting larger, and so the whole tumor is getting larger. And we start seeing cell division and so on. One thing that I have noted here is that after I did this change in rehabilitated growth, I changed the threshold a little bit because we see that the proliferating layer is actually quite thin. It's only 1 cell deep, and here for a while it wasn't even 1 cell deep. So what I'm going to do here is I'm going to change my PQ to be 80% of what it was. So I'm just going to multiply everything by 0.8. And yeah, I'm definitely going to need more than 10 minutes. So now we have growth again, and again, I changed the threshold here. And again, the code that I uploaded looks a bit different, but it's not. It just looks different. And actually, it's the code that we see here on my presentation. But what's the difference really? We have a if statement, if a concentration, which is here and here, it's the same number, is less than a number that in that code there is saved in the self PQ handle, but here it's saved inside the dictionary do something. So it's really the same code. And we can see that since we have growth, but our necrotic cells aren't really dying, the whole space fills up. And because this version doesn't have the slightly adjusted threshold for quiescence, the proliferating layer is a bit thin. So now we see that the proliferating layer is way thicker. It's 3 cells, which is about what we wanted before. But okay, we are telling cells to become necrotic, but we are not actually doing anything with that. We have to actually, well, if I just let this run for a while, we're going to see the 3 layers again. But the central layer just stays there, chilling instead of dying. So we have a few options on how to kill our necrotic cells. We have 4 options, but I'm only gonna tell you of 3 of those. You can see the third here, I think. No, here. In the cell manipulation menu. We can either set their target volume to be 0. In that case, from the point of view of the algorithm, the necrotic cells are gonna pull everything in. Or we could set their lambda volume to be 0. In that case, the necrotic cells would behave like a foam and would slowly peter out. Or we could slowly decrease the necrotic cells target volume. And what we are going to do is set their target volume to 0, because this is way more fun than this. So here is lambda 0. We see that the tumor eventually invades everything, but it's kind of boring. While here with the target volume 0, we have this interesting pulsating behavior. Are you sure you are trying to increase the target volume instead of the volume?
Can we freeze them?
We could, but I mean, the necrotic cells should, I guess, disappear. Yep, if it's frozen, they won't disappear. So here, when they become necrotic, what I'm going to do is I'm gonna copy this line over, but instead of growing them by 0.5, I'm just gonna set that to 0. And it should result in something cool like this. So if I stop this, restart, and we go. Still in the growth regime. Cell adhesion still applies. So if if we look here, not in the steppable, you'd need to create a cell type that is frozen and change to that. Here we have necrotic contact energies, quiescent necrotic, and so on. So we see that indeed the necrotic cells shrink, but the simulation is kind of slow to run. That's why I have a ton of GIFs here on the left. So cool, now we have a vascular tumor that has the 3 layers and behaves somewhat realistically. But we are not actually, the cells aren't actually feeding, they are not consuming from the nutrient. Nutrient simply decaying inside of them. So we could instead uptake from the field and use that for growth and to set the transitions from one type to the next. But there is one thing about uptake that I think Josh mentioned yesterday, that we could naively assume that we could just take some amount from the field. But that has problems because computers are very literal. Some could say dumb. And if you try to take out 10 units out of a concentration of 1 unit, you end up with negative concentrations. So what is usually done is that we take a percentage of the available concentration instead of just a number straight up. And in Compucell, by default, all of our uptake functions are of this form. So it takes a percentage. So not only it takes a percentage of the concentration, but there's also a upper limit of concentration you can uptake each time step, which again makes sense. Cells take time to get the nutrient in and actually send it to the mitochondria and do stuff with the molecules. And again, I think Josh mentioned yesterday, we have several options of uptaking just outside the cell, just inside the cell, in the center of mass of the cell, over the whole cell. We're gonna do over the whole cell. But I think this is a good point to add a pause, and we can carry on from here after lunch. So I'm gonna bring back the code. Yep, I'll upload this to that drive right now before I forget. So one nice thing of Twedit is that it also knows how to zip your project. Then to open Jupyter, this might take a little while to open. Let me stop the share here. Oh, a quick parenthesis as well. So one nice thing about using Medium as a supplier is that we could calculate what is the tumor size limit based on the size of the lattice and the size of the tumor. Yes, we are done for now. We are gonna carry on by changing the way our tumor behaves to be based on how much nutrient it consumes after lunch. So I'll see you all in 25 minutes, give or take. So at 2:15 Indiana time.
Please don't forget to download the demos of module 3.3. I post the link on the chat.
Pedro, why don't you leave that slide up as the slide you just showed up on the screen as a screen share?
You mean the first slide of the presentation?
For the next one, just saying when we start again. That's perfect. Thank you.
Hello again, everyone. Welcome back. We are gonna give people a few more minutes to finish up their lunch, go to the restroom, etc. So we should start at 2:20.
Make sure you downloaded the demos on module 3.3 and upload in NanoHub if you're using a NanoHub.
Yep. And also the avascular tumor, the completed code that is a bit different from what I did in classes there, and what I did in class so far is also there. And just a reminder, we are being recorded and live streamed to YouTube. Okay, so it's 2:20 right now in Indiana. Once again, I should remind everyone that we are being both recorded and live streamed to YouTube. And we are gonna get back into module 3.2, part 2 of it. Part 1, we've built that simulation there that there's something about it that just looks nasty to me. I really like it. And what we did before was we changed cell behaviors based on the measured concentration. And now we are gonna have our tumor cells actually eat up from this nutrient field here, instead of just letting it decay inside of the cells. Okay, so getting started. Let me actually go back a bit on what we did. So what we did here in the steppables was we have here the constraint initializer, where I'm initializing the threshold numbers. The proliferating quiescent transition, right now I'm setting it to 80% of 100 divided by half of V. The quiescent back to proliferating, I set it to be just a bit higher than the other one. And finally, the transition to necrosis is 100 divided by E. I think if you disable the cell borders, it's disabled on all of the visualizations, unfortunately. We've also set ourselves to be 64 voxels big with a lambda volume of 8. And then here in the growth steppable, we are doing all of the magic. We are telling CompuCell to look for this cell dictionary called conc to make it easier to visualize what concentration we are measuring inside of the cell. So it looks similar to the nutrient field, but it's clearly different. It's discrete, while this one is smooth. And then in the step function, we are asking for all of the values of the nutrient field. Then we are going over all of the proliferating cells. We are measuring the concentration at their center of mass. We are saving that to the cell dictionary. That's what allows us to see this field here on the right. And then we are checking if that concentration is below our transition threshold for quiescence. And if it is, the cell becomes quiescent. If it's not, it's still proliferating, so it grows by a little bit. And then we are doing something similar for the quiescent cells. Again, measuring the concentration, saving it. If the concentration is above the threshold to go back to being proliferating and becomes proliferating again, and it also grows by a little bit. And if the concentration is below the necrosis threshold, we are setting them to be necrotic, and we are setting their target volume to be 0. So here we have less than, because we are transitioning from proliferating to quiescent, and proliferating is more demanding on the cell, so they need more nutrient. So it makes sense that when it goes down in concentration, we go to quiescence. While here we are asking if we can go back up to be proliferating. So that's the switch here. And now what we are going to do is, instead of using the measurement at the center of the cell, we are going to have the cell's uptake from the nutrient field, and we are going to use that as a basis. So, as usual, Twedit knows how. Although in just a minute, Twedit won't know how. But that's spoilers. So what I'm going to do is I'm going to come here before my first loop. And I want to do uptake. So here in CCD Python, we have secretion uptake. Uptake is negative secretion from the point of view of the computer. And I'm going to uptake inside the whole cell. So again, before the loop, CC3 Python, secretion uptake, uptake inside cell. And now we have the commands we need. So first, it's creating this object secretor, and that object is responsible for the uptake. And we can see that to have the cell uptake, we call this function of secretor passing to it a cell, the maximum uptake the cell can make, and the relative uptake. So this function here, as a reminder, looks like this. So while the concentration is above this maximum, it's only going to take the maximum. And if it's below the maximum, it's going to take a percentage of the concentration that is there. But here we don't, well, if we try to run, we would get a complaint of cell used before assignment, because, well, there's nothing here saying cell equals something, while here we have. And one point that I'm going to make is that the name cell here is not important. I could very well have ASD in cell list, and ASD would still be cell objects. After all, we are iterating the cell list. This handle here is important. So for this loop, I'm gonna make my life harder. Don't do this. And have ASD instead of cell. But again, don't do this. I'm just making my life harder because I don't know. And so what I'm going to do is have the secretor function be inside of the loop over the cells that we want to do. But I actually need to specify what field I am talking about. And the field is nutrient, which I called NUTR. And then I'm gonna cut this command here to be inside of the loop. And indentation matters, so putting that in. And on my slides, I have that I set the max uptake to be 1 and the relative to be 0.1. So I'm gonna keep that. And in here, so if I try to run this, I'm gonna get an error right here. Just to prove it to you, I stop it here, play. There you go. But if I do this ASD now, things should be good, hopefully. Otherwise, I'll have to eat my hat. And now the proliferating cells are going to line 56. Yeah. So yeah, line 56, I had cell instead of ASD. And that's where it complained. Now I have ASD, line 56. And it works. But I don't want only the proliferating cells to be eating up nutrient. I also want the quiescent cells to be eating up nutrient. So I'm just going to copy over that command to be down here. And in here, it should be cell for me. I'm not changing this cell to be ASD, and please do not change any cell to be ASD on your code. Now we have both cells eating up nutrient. This does mean that the chemical change profile is going to be different from before. Before we were only letting the chemical degrade inside of the cells. Now we are actually taking out of the chemical food inside of the cells. So we need to adjust our thresholds again. And this would be kind of trial and error. Thankfully for you, I already did the trial and error, so I know what to do now. I mean, we could leave it as is, but I think it becomes more interesting if we now change, instead of having 80% here, we're gonna do 50% of that number on the right, and we are also gonna diminish the necrotic threshold by 50%. So again, me just gonna let this run while I talk. What I did was first asked to edit how to do secretion. I came before my loops, went to CCG Python, secretion uptake inside cell, the whole cell. It pasted both this command, but without the field name there. I put that in, and it also pasted this command with some other numbers there. So I moved the command from back here to be inside of the relevant loop. I set the max to be 1, the relative to be 0.1, and I also have the same command down here. Okay, so now how do we visualize how much the cells are actually eating? Well, this is one thing that Reddit doesn't know how, for some reason. It's a simple change to this uptake function. We just need to add total count. And if you notice here, the first letter of each word besides the first is capitalized. This is called camel case. And different programming languages have different standards for when to use camel case and when to use snake case, which is using underscores to separate words. And in this case, it's camel case, so we add total count like this to the end of the simulation. And we actually need to get the results from that function. So I'm gonna have R equals secretor dot uptake inside cell total count cell max relative. And I'm gonna have the same thing for my quiescent cells. And now to visualize this, I'm gonna replace this extra field that we created before. Instead of having the concentration at the center of mass, I'm gonna place this before we actually do the uptake. And what I want is the results from the uptake. So I'm gonna replace the concentration at COM by R dot amount. And I'm going to do the same thing down here. There we go. So now if I stop this and rerun and we go. I change this to be that conc field that we had. And I keep this with the nutrient and run. What is going on? That I didn't expect. What did I do wrong? Yes. Comment out the uptake. I have an idea of what's going on. Right. I shouldn't have replaced the value here at conc. I should leave that as concentration at center of mass, because I'm using conc here to chain for the thresholds. And so instead of leave this here And what I'm going to do instead is I'm going to create a new cell dictionary, and I'm going to call it something else. I'm going to call it for uptake. And this one, I'm going to have r.total count. And same thing down here. And then to actually have Compartmental track this, just need to copy this command and tell it to search for UPT. So I asked to edit how to do the uptake. Then I told that I'm looking for the secretor object for the nutrient field. And in my proliferating and quiescence loops, I have here the result of the secretion saved to R, and the secretion is being done inside of the whole cell with a maximum of 1 and a relative uptake of 0.1. And then I'm putting the actual result, which is not R, but R dot total count, dot count, to this new cell dictionary, UPT. Now I go. It's not right. I should read my slides. It's not dot count, it's dot amount. And now if I tell it to run, it should work. Good. So if I change this field here to be UPT, we can see that all the numbers are very negative because uptake is defined, as far as the computer is concerned, as the negative of the secretion. That's why we have the minus. And one thing that is not that noticeable here using the extra field, but is noticeable if you print the result straight to console, is that the uptake is about the cell volume. Because uptake inside cell total count, this maximum and relative. So for the secretion, I would point you to the manual. And I had total, its total amount period. My brain got confused. But the result is about the cell volume because this maximum and relative is for each pixel of the cell. And since we are telling it to have a maximum uptake of 1, the maximum uptake result is the cell volume. And that gives us an easy way to normalize the uptake based on cell volume and so on. Yeah, here I have it. So before changing, so the next step here would be to change the growth based on this uptake. But before we do that, I actually want to change this ifs to be based on the uptake instead of being based on the concentration at the cell center. So thankfully, with the parameters that I used, the result of the uptake is going to be the cell volume. So what I'm going to say conc is is instead of being the result total amount, it's going to be uptake is negative because it's reverse of secretion, and secretion is positive. It's going to be the absolute value of the result. That way you get rid of the negative. And I'm going to divide that by the cell volume. And that way, this number here, which is the concentration at the center, is going to be similar to this number here, because this number here is the average concentration of the cell, while here is the concentration in a single point of the cell. So they should be about the same. And I have to do the same change down here in this other conch with my quiescent cells. And if I run this, I'm gonna get an error because I made my life harder by changing cell here to be AST. So there, absolute, for the absolute value of the number. I could, instead of doing abs, I could have done just the negative of the number. In this case, it's the same thing, but yeah, good catch, man. So hopefully now things should still behave more or less the same. Probably. We're gonna know in a second if I have that mistake. Yeah, okay. Have something wrong. Joseph might know already what I did. Yes. Yeah, so this should be down here. And it still may not work because I didn't test this before. So live coding is always a trio and things would never go right. So if this doesn't work, I'm just gonna roll back the changes and use the central, the central value. Not really sure why this didn't work, but I'm not gonna waste time from the class with something that doesn't work. So sorry to make you mess around with your code and then undo everything you did, but keep the cell dict count to be the concentration at the center of mass. But if you wanted to change the thresholds to be something related with how much the cell could eat up, that's the general idea. You'd need to fiddle with the thresholds and check what exactly are the results of the uptake and so on. And doing that, it's easier on the local installation because here we don't have the terminal, while in my, if you use Compucell locally, it's going to open a terminal window that has all the prints. Yes, if you wanted plus before. Not sure I understood the solution, but if you might try it. Plus before. Ah, okay. So keep updating the concentration. Yeah, maybe. But I'm gonna keep things working. So the actual change we are going to do is we're gonna change the growth. Instead of being a fixed amount, it's going to be a part of the nutrient. So I'm assuming that part of the nutrient that the cell consumed is used to keep the cell alive, and any surplus it has can be used for growing. So here in my loop over the proliferating cells, in the else, which means that the cell is still proliferating, I'm gonna, instead of hitting plus 0.5, I'm gonna have plus 0.5 times the absolute value of the uptake amount divided by the cell volume. And as I mentioned, this number here is going to be about right. Nice. Good. Glad that the solution was easy. And I have to remember to do the same down here. So now, if I run this, yep. Keep forgetting that I'm making my life harder. This is why I told you not to do this. But I thought it was an important point to make, that cell is not special. You could do ASD, you could do flower, you could say cat. And cool, the simulation still works. It actually looks remarkably similar to before, but that's kind of part of the point. And the next exercise is doing what tumors do best, which is mutating. In the slides and the code, full code that I uploaded, I'm mutating a lot of stuff of every cell. I'm letting every cell mutate their thresholds, so they could be more resilient to lack of nutrients. I'm mutating how much they eat, so a cell that eats more has a competitive advantage. I'm changing how much they grow. A cell that grows faster also is more competitive. But in class, I'm just gonna mutate one of their attributes. That way we can see more fun things. And the attribute that I'm gonna mutate is growth. So the first thing is, instead of having growth be this hard-coded number here, what I'm going to do is come to the very start of the step function, and I'm gonna re-enable this loop that I disabled way back at the start of the simulation. But I don't want the cells to grow here, so I'm gonna get rid of that, or just keep that part commented out. And I'm gonna say that every cell now is gonna have a dictionary attribute called growth, which is how fast it can grow. And I'm gonna set that to be 0.5. And since I'm already here at the top and I'm near the init function, I'm gonna create one more extra field. And this one, Compartmental is gonna look for the keyword growth. And so instead of hard coding, well, it's still hard coding and it's here, but this loop, the way it is gonna nullify any mutation, because for every step, I'm going through all of the cells and resetting their growth to be 0.5. So I only really want this to happen in the first MCS. So I have two options for MCS 0. I could either say if MCS equals 0 and put this loop inside of the if, or I could remember that 0 is equivalent to false in Python and say if not MCS. Either works. To me, they are both equally clear, but personal preference. So if MCS is 0, set that growth. And now what we are going to do, and we are almost done, is, well, first, remember to replace the halves here by self-dict growth. And then we are going to have mutation happen when the cells divide. So we are going to go all the way down to the mitosis steples that we forgot about until now. So after clone parent to child, we are going to have our cells have a really high mutation chance. We are going to have a mutation chance of 90%. So if numpy that I've imported already, dot random, dot uniform, because I want uniform distribution between 0 and 1, is less than 0.4, this gives us a 90% mutation rate. Then I want the cell dict, the growth dict, which is here, to change by, well, in the slides I'm saying plus or minus 10%, but let's be more aggressive just to make things more visual. So cell growth is going to be multiplied by, well, it's going to equal itself multiplied by numpy.random.uniform again. But in this case, inside of uniform, gonna change stuff by plus or minus 50%. So uniform from 0.05 to 1.5. Well, and here it shouldn't be cell. It should be parent cell for the parent cell. And then I'm gonna copy the whole if, and the child cell can mutate independently of the parent by a different amount, and it may not even mutate. And this is pretty much it for the lecture. Now you know how to do a vascular tumor and how to do cancer mutation. What else could you want? Cell reference before assignment. Did I forget that I was using ASD? Yes, I did, as usual. Let me change this one to be growth. And for now, everything is in half because the cells haven't had a chance to mutate yet. But as soon as they mutate, we are gonna see this one. Well, actually, let me do different. Let me change this to the cell field and this to the growth field. And we already have cells that are growing faster than others. And we are probably gonna see those cells surviving more and become the dominant strain in the simulation. Yep. Let me go back to the code. So I went all the way down to the mitosis stepable in the update attributes. After we clone everything from the parent to the child, I'm giving both the parent a 90% chance of mutation and the child a 90% chance of mutation. Well, each of the siblings really, right? So rolling a dice between 0 and 1, and if that's less than 0.9, then changing the growth by multiplying it by some random number in between half and 1.5. And same thing for the sibling. That's it for me. And I'm going to upload this to the Google Drive as well in just a second. James, back to you.
Gilberto, are you ready? Gilberto, you okay? There we go.
We are not hearing you, Gilberto. Check your settings.
And now?
Yes, yes, we hear you now.
Can you see my screen?
Yes.
Okay. Let me start. Okay, good afternoon, guys. I am Gilberto Lima Thomas. I am a professor here in Federal University of Rio Grande do Sul in Porto Alegre, Brazil. I collaborated with Compartmental people from a long time, maybe 20 years already. And today I'm going to talk to you about this cell crawling demo, which is a more simple model from a published paper we have done last year. Okay, I will start with this picture here, where I include the topics that I will cover in this talk. I will start with talking about cells in movement, talking about some biological aspects of the cells in movement. And in this demo, we will try to implement some functionalities of the Compartmental 3D, which are here listed: contact energy, graphical plots, how to output data, how to include the diffusion solver, a diffusion equation solver, a hint on Chemotaxis plugin, compartmental cells, and how to fine-tune parameters, the steering panel. Motivation, contents. Motivation, I will speak about why we are studying cell movement. I will put some main goals of this talk, and then I will start to build a computational model in 2 dimensions. In this part of the talk, I will build the code step by step. First, the first part, I will make the single cell move. Then I will include some plot and data saving features. A student of mine, a grad student, will present a demo where the single model can be expanded to include multiple cells. And finally, I will left you a homework on where we are going to include the steering panel to adjust the parameters of the simulation. The solution of this exercise is already uploaded in the students' material folder in the Compartmental workshop website. Okay. Well, starting. Motivation. The basic question we are trying to answer: why do living cells, isolated or collectively, move around within their environment? Well, this is a big question. Right? So we do know that cells crawling and migration is ubiquitous in many biological processes. For example, I can talk in the embryonic development, or for example, here we have a simple, well, simple is a matter of speak, but a complicated the process of gastrulation in the early embryo development, where the cells move to form the gastrula, invaginations, a lot of steps in this process. Wound healing that it was presented already in a demo yesterday, I think. Also, we have the inflammatory response when you have infection in your body, like the COVID-1, for example. We have this response from the immune system, our defenses, with the cells moving to seek the invaders and to kill them. And we have, for example, cancer metastasis, where the cancer cells flow through the blood vessels, and somewhere in a blood vessel, they go out of the blood vessel and start another cancer focus in the body. Okay, so there is many reasons to start to study cells. How do the cells crawl, move? This is the motivation. We know that a single cell presents a different movement mechanisms. Those mechanisms can be either external mechanisms, for example, this flagellar mechanism, or a cilium mechanism in a spermatozoid here, or for example, a paramecium here. Those are external mechanisms that the cell can move, one single cell. Also, we have internal mechanisms that make the cell move. The first one, for example, is the lobopodial motion, where the cells make some tubes, some structures like tubes, and pull itself forwards. Another motion, another kind of motion is the amoeboid motion. Is this one here. You can see the cell moving like an amoeba and going after some food, some another bacteria or something. It's moved this way, like a whole body. And finally, the lamellipodial motion, and the cell make a protrusion with part of it. This protrusion advances the stick and pulls back the rear part of the cell and move around. This is the part, this is the mechanism we want to study right now. Okay. So, how does it happen? When the cell moves in this way, he can move with different behaviors. In this small movie here, you can see it's not the best movie I found, but anyway, it's here. The first part of move, this one, is only a diffusive motion. The cell practically doesn't move around. This just randomly moves without go anywhere. The second movement is the persistent movement. The cells goes, the cell goes in a direction quickly. And the third one is the mixed movement. There is a part diffusive, a part persistent, and those are the three different behaviors during this kind of motion. The engine of this lamellipodial movement is a complicated actin network inside the cell. When we have the cell has some bunch of monomers of actin. These monomers arrange themselves in fibers. And these fibers, as you can see in this fluorescence image here, make the protuberance of the leading edge of the cells, and the cells advance and pull back the rest of the cells towards this front here. Okay. In the edging, the border edge of the cell, the monomers have a mechanism like a treadmill mechanism. The monomers are putting together in the border of the cell here, and at the end they are disassembling. So the part which is disassembling here goes to the front and push back the border edge towards. Here in this small move, you can see this thing happens here. Okay, this is the way the cell goes forward. The monomers are coming from here to there and forcing the leading edge moving that direction. So this is the kind of motion we want to reproduce in our simulation, in our model. There. So the main goals for this demo is to reproduce the single cell random movement. Then this is not enough. We have to plot the cell trajectory and to record the data we have from the simulation to be able to compare with experiments. We are not doing simulations just for the pleasure to do it. We want to do the simulations to see if we can reproduce reality, reproduce nature. So we must be able to compare our results with experimental results. So we have to save the data, okay? After that, we will try to extend this single cell demo to multiple cells. Then why? Because we want to study after collective model, motion, for example. How do the wound healing happens, for example, in a tissue motion, okay? And after the parameter adjustments, I mean, to include how to include the steering panel in our simulation to fine-tune the parameters to make our simulation more realistic, to reproduce the real cell movement. Okay, let's start talking about what biological model we are going to follow. We are going to have as a hallmark for building our in silico model. Okay, so we are going to follow the Abercrombie model. This Abercrombie model is the cells moves this way. The cells make a protrusion, which is the lamellipodium. The lamellipodium sticks to substrates. Then it pulls back all the rear part of the cell, detaches the rear part of the cell from the substrate, and moves forward, starting again the cycle. Okay, so in this model we have basically these components here. We have the medium, which is this place where the cell is living. The substratum is where the material where the cell adheres to pull back, to pull itself. The cell proper, the proper cell constituted by three parts: the nucleus, the cytoplasm, and the lamellipodium. And we have this actin cortex, which is that network of the fibers that I showed before in the fluorescence picture, and the focal contacts. The focal contacts are the adhesion between the cell and the substrate. Okay, this is the basic model we will follow. Then the computational 2D model now. How do we design this 2D model? It's quite simple. It's simpler than the previous slide. In this 2D model, we are going to have only the cell formed by 3 components: nucleus, cytoplasm, and lamellipodium. We need those to provoke the motion to induce the motion in the cell, the medium where the cell is, and the actin cortex. This actin cortex will be emulated by a chemical field, which will coincide with the lamellipodium. See, here is the chemical field that we are calling F-actin field, because it is supposed to emulate the actin network fibers, monomers. So it coincides with the lamellipodium in green here. It is collocated underneath in the simulation. So we have only these 4 components in our model. We don't have the focal adhesion points where the cell is stick to the substrate, neither the substrate itself. Okay. So let's talk about the dynamics of this model. How the cell will move with this model. So the cell is here drawn in the grid. Okay, the small squares represents the grid, the pixels in the grid. Okay, so in this first orange window here, we have a pixel, a white pixel, and which is the lamellipodium being copied over the medium. Why? Because we have this actin field underneath the green part, which in the calculation of the energy variation during the Monte Carlo step, as you know, this actin fields favors the copy of the lamellipodium over the medium. So, okay? So if there is a attempt to copy a green over a black, this attempt will be favored by the presence of the F-actin field underneath the green lamellipodium. Well, if the copy is favored, this will increase the volume of the lamellipodium, which has a target volume. I mean, if the copy is favored, the volume will increase above the target volume. So when you have now an attempt of copy from the cytoplasm in blue here over the lamellipodium in green, this copy will be favored because the target volume of or better, the volume of the lamellipodium is above the target. So the copy will be favored from the black, the white in the cytoplasm to the gray, to the red and green. So in this way, again, we have an increase of the volume of the cytoplasm above the target volume. The last step of this process is, well, now I have the cytoplasm above the target, so it favors that a medium pixel copies itself over the cytoplasm blue. Okay, then green goes to black, blue goes to green, black goes to blue. So we have then a process that involves 3 steps, creating a polarization axis for this motion. And the cell as a whole moves in that direction by itself, only because we have this initial chemical fields emulated the actin fibers underneath this lamellipodium parts of the cell. Okay, this is the dynamics that the model proposes. We have here the computational model, the physical part. I think you have done enough with this part. The energy of the system at each Monte Carlo step is given by this equation here, where this is the interface energy between different types of components in your simulation. The target volume, which prevents that the volume of the component goes much higher or much lower than the target volume. And this effective energy, which has only sense when you calculate a difference between this energy. And this difference is written in such a way that only this difference is only 0 if we are make the calculation between lamellipodium and medium. Otherwise, this is 0 and doesn't contribute for energy at all. Okay? The actin field here, in principle, is solved by the diffusion equation solver. I mean, it's a diffusion equation here where we have the time of variation of the field, and you have the Laplacian here of the field, the source term, and the K term of the field. This is a usual diffusion equation for the effective field. The last part of this model is really we have to include the diffusion folder, the solver, but we don't have to use it right now. Going ahead, let's start to write the code. Now you can open to edit. Okay. Remember that the simulation has these components: medium, nucleus, cytoplasm, lamellipodium, and actin cortex emulated by the actin fields. According with the simple model, we are going to build our initial state for the simulation like this: 3 square cell types, each one with one of the components of the cell, cyto, lamellipodium, and nucleus. The colors are the same as I used in the previous slide. And the dynamic is the one I just talked about. Okay, so let's go to the edit and let's do this as exercise. The exercise is the code is already loaded in the student materials. You can upload that to the NanoHub if you want, but let's try to build the code step by step to understand what we are doing here. So open to edit, create a new project here. The simulation wizard will start. You put name your simulation, cell MIG 2D. You choose a destination for where your simulation is going to be saved, and choose here the simulation type, which has Python codes, Python files, and XML file. Click next, and at the same time, your simulation, your wizard will create this folder. Oh, sorry. will create the folder you just created here in the path you gave to it. Now, the next window in the wizard are the parameters of the simulation. The property, the general properties like let's dimensions. We have only X and Y dimensions. We are doing a 2D simulations. And we want the boundary conditions of the simulation to be periodical, because we are simulating a cell that is moving on an infinite plane, if you want to. So the cell can goes in a left direction, and if the cells go outside the grid, the cell will appear to the right side of the simulation, as it was moving in a tire, in a torus. The simulation, the grid acts like if the cell is moving off a donut, if you want. Okay, there is no boundaries to the motion. This part you let as is. Don't change anything. Let's type, not in situations, blah blah blah. Number of Monte Carlo steps, and you will start with rectangular slab. Click next, and add the specifications of the cell types you want to include. Medium is already there. You include the cycle, the lamel, and the nucleus, and move forward. Next. No, don't forget you have to add it. It's a cell type you write here. You have to click on the add button to include in the list, in the table. So go. The chemical fields. Well, we'll have only the one chemical field here, that is the F-actin field. F-actin field, which will emulate the actin network. Click the name of the F-actin field here, and choose one of the solvers here, which is the diffusion, flexible diffusion solver FE, finite elements. Click and add, and next. When you click add, the field, the name of the field will appear in this table as well. The next wizard, the next window wizard is the properties and behaviors. You have to choose contact energy, Chemotaxis plugin, volume flex plugin, because we want to identify each component of our cells separately. Okay. Then center of mass is already there, and you have to choose this next one, which is the cell pixel tracker. Cell pixel tracker. It's a plugin that allows you to pick a site, a pixel on your grid, and determine if to whom that pixel belongs, to which cell the pixel belongs. Okay, so click next, and you go to the Chemotaxis plugin. You have to configure the Chemotaxis plugin. This is the dynamic part of the code. Because, see, here we have to say, where is who is producing the F-actin field? So here is the lamel, because the field is created underneath the lamel. So cell type lamel. Here you put a value lambda, which is the strength of how the cell respond to the chemotaxic field. If positive, the cell will move towards the gradient. I mean, the cell moves searching for a high gradient of the field. If it's negative, the cell tends to escape from to move from the higher gradient to the lower gradient, towards the lower gradient. Okay. And here is the trick part, say chemotaxis, lamel through the medium. So the lamellipodium must move in direction to the medium. This is where we create that dynamics. Okay, add the entry here and click next. Are you following? Should I wait a little bit for this part? I think everything is doing well. Okay, let's go. So, next part. What happened here? The next part, you have complete your configuration. So, click Finish. And the simulation wizard closes, and your project appears at the left side of the toolkit. If you click on it, the simulation, the tree expands here, showing you the three created files: the Python file, the XML file, and the steppable file. Now we have to start to adjust the simulation. I mean, when you click on the XML tab here, you can see the values that the wizard have configured. Sorry, let me go back. So those are the values that come from with the wizard. This first part of the XML, it's okay, you don't have to do anything. Here is the size of the grid, 128, this number of steps, the fluctuation, boundary conditions, cell types, plugin volume, plugin center of mass, and plugin pixel tracker. The next part of this file, we have to start to change, to adjust. Those are the values that come with the contact plugin. Everything comes with 10 as a value for the contact energy. So we don't want this value. We want our cell to have this kind of shape at the end. So we have to adjust these values such that the cell has this shape. I mean, nucleus must be inside the cytoplasm, surrounded by the cytoplasm, and cytoplasm that must be surrounded by lamellipodium. So we have to adjust these energy values here. And this is done using these values here. I think I'm going to copy these values here and put in the chat window. Then you don't have to type everything where is the chat window? I lost the chat window. Here. Okay. So you copy this part of the chat window and sticking the two over the old one into it. Then you have the correct values for the correct shape of the cell at the end. Next fix we have to do is to change something in the well, the next plugin is the Chemotaxis one. The Chemotaxis one is already adjusted within the wizard. It has the correct towards motion here, major, medium, lambda 100, and as you can see in the simulation, in your simulation, it will appear the cell type lamel there. And the next part, the steppables. After finishing with the Chemotaxis plugin, you have the steppables. The steppables, the first steppable is the flexible diffusion solver FE that we include in the wizard. It comes with a bunch of values here as default. Diffusion constant, decay constant, blah, 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 some comment lines that you may need in some kind of simulations, and some consideration of boundary conditions. Bottom line, you don't need any of those lines here. So you can erase everything but the field name tag, this one. Just keep in your simulation the field name active. In this way, we are using the diffusion solver here just to include the active field in your simulation. You are creating the field, but you don't need to solve it right now. We are going to solve it in a trick way later. Okay. We are good. Can I go ahead? I think it's no complaints at all. Good. Last steppable, the uniform initializer here. As default.
Sorry, Gilberto,
Yes.
use the poll. Are you done with the exercise? I think one or two people are saying that they'd like a little more time.
Okay, thank you. I will wait a little bit then.
If you go to the bottom of the screen where the controls are, one of the options is polls. And
Yes.
that will show you the
Yes. Sometimes I get confused here because I'm using two screens, and so I have to go from one to another, and
So there's a request to show the previous slide.
previous slide. This one. This is one where I was talking about to erase everything from the diffusion solver steppable, keeping only the field name, the diffusion data field name tag. After that, I came to the steppable uniform initializer. The boundary conditions, yes, you should raise the boundary condition. The only tag that is kept is the field name. Okay, I think I can go down. Okay, this uniform steppable here, the uniform initialized steppable as a default came in this way, where we have here the three created cells, cyto, lamel, and nucleus, disposed, occupying a big part of the grid. But we want a specific initial state that is that only three cells disposed, for example, this way. So we have to rewrite the uniform initializer this way here, that I also will copy from my code, and I will put in this in the chat window. You can copy and paste in your simulation. It's much more simple than type everything again. See, in this, we have the three regions. In each region, I have only one cell, cyto, with size 9. I will square with size 9, disposed like a L here. Okay? And now, okay, I think I can go ahead. Okay, once you have done that, you are done with the XML file, you move to the Python file, the first one. You don't have to do anything here for now. It is defined by the wizard, and you leave as is for a while. Finally, we now move to the steppable file. The steppable file also comes with these default lines. Basically nothing here, only this line here in the step that prints the cell ID. But now we have to give the cells we created some signs. Because remember, we create our cells in the XML file with no attributes. We only load the volume plugin and the center of mass plugin, but we don't say anything about how big are the cells and how is their target volumes. We need to do this here in the start function. Okay, so in the start function, we are going to include these lines here. Again, from my simulation, I copy just to help you guys and put in this chat window. See, what I'm doing here is I make a loop. I run over all the cells in the simulation and say if the cell is type cyto or cytoplasm, I give them the target volume of 300 and a lambda volume of 10. For the lamel, a target volume of 100, a lambda volume 10, and for the nucleus 50 and 10 again. Can someone post the code? Which code? The one I am writing? Some people The code. Pedro, can you answer Tarim? Okay, thank you. I don't know if this is the code he wants. I think he wants the whole code in the student's material, maybe. Okay. Continuing, let's see. So when I include this in the start part of the steppable file, now I need to create the actin field. Remember, we just use the diffusion solver to create the object actin field, but we don't have it in the simulation yet. So we need to create it. This is done by here in the step function. By each time that the simulation entering this code, it will the lamellipodium will create actin field underneath it. In this way. Here's the place. See, I will create a variable here, which is where I will put the effectin field, self field effectin. It's an array in 3D with only one size in the Z direction and 3 in the XY size. And make a loop in this cell list by typing, saying that for pixel belonging to the lamel cell, I will say that the field underneath that pixel is 1. I'm saying that the field underneath the lamel has the value 1 and is 0 everywhere. So the field is now a binary field. Is 1 underneath the lamellipodium and 0 everywhere. Okay, so there is a gradient between a gradient in the field between the region underneath the lamellipodium and the region outside of it everywhere. Since our lambda is negative, the cell will tend to move in the direction of the lower value of the field. It means to the medium, moving to the medium. Okay? This is the dynamics of the motion. The polarization axis is created here. Right? So let me copy these lines to put inside the step function and erase that loop where you are printing the cell ID. You don't need that. So finally, this is how the steppable file looks like at the end. We have at the start function the cell target volume and lambda volume being given to the cell types, to the different cell types. And in the step function, the place where the field is created every time, every Monte Carlo step. So once you have this completed, save your project, click on the double diskette here. and you are ready to run your simulation. The best thing to do now is to you click on the CC3D logo here, or alternatively, you can right-click on the cell project, cell MyGit2D point, CC3D logo, project name here, and say open in player if you want. I have it here, I think. Here, if you click here, open in player, and the simulation will be loaded in the player. If you click on the first time in the CC3D logo, the player opens here, and you have to go to your simulation place for the first time, open simulation file. You have here, let's see what is this, here, and run. You have your cell moving around. See, this is what happens when we don't have it fine parameter, fine tuning your parameter. This is perfect well right now because you have to adjust. This is the first stage. You create your simulation and your cell is moving. Okay, I have here a small move. Here the player open, go down, down. Here the initial. If you click from the beginning, you will see your initial state in the player. Click here, one step here. You have your simulation. The initial state built in that way. You click one, run, and the cell will move. See, we don't have, here we have a emulated cell, which is built with 3 different cells. The shape of this moving cell here, that we can call a cell moving here, is that way because we have chosen the right contact energies between the 3 different types of the cells. Okay, we have a right energy between nucleus and cytoplasm, the right energy between cytoplasm and lamellipodium, and the right energies between everything else and the medium. Okay, so you have your simulation done. A very simple model where you have a moving cell, very similar to the ones you can see in experimental setups. Let's see if there is some error here. I'm gonna open the poll here to see if you people.
Gilberto,
Yes.
we probably need to take a break.
Yeah, sure.
So we can talk over the break if you like, but we should have a break until 4 o'clock.
Okay, I can stop here now. This is the
And then,
end of the first part.
Okay, perfect. And then start again at 4 o'clock.
Okay.
So if people have any questions, please ask, and please fill out your response for the questionnaire for this module.
Okay. We are back. Now I'm going to start the second part of this talk. Actually, the second part are more, we have more exercise to do, okay? The first one is the following. Remember I talked at the beginning, we want to be able to see the trajectory and to plot and record the data which is being created during the simulation. Okay, this is our task now, to plot the nucleus trajectory, because usually in the experiments, the nucleus is the best part to follow during the experiment. It can be easily colored by some dye, and with some fluorescence microscope, you can follow during its movement. Okay? So let's follow the nucleus and record the data. So now we can to not change the simulation you just created. We uploaded an extra simulation in the students folder, which is called something cell MIG 2D plot and rec, something like that. This is the one we are going to use right now. But I'm going to show you here how to change the previous one in the second one. So you have your previous simulation open in the Twilio here, and go directly to the steppables file. Locate your mouse in some empty part, and go to the CC3D Python tab, scientific plots, setup. This will create a plot window in your simulation like this. Set plot window, blah, 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 blah. It comes with some default lines here, with title in the axis, title in the window, and which plots I've been included in the graphing. Here, for example, we have the volume and the surface of a cell if the cell is in 3D. But we need to edit this one. We want to plot the nucleus center of mass. We want to follow the center of mass. So we have to change these lines to say that to the code. And this is done. Editing the title, nucleus CM trajectory, x-axis, put the x position of the center of mass, and the y-axis, the YCM, the position of the center of mass. And call this window, give it a name, nucleus CM. And we are going to plot it with lines, color red, size 5. I can copy these lines for you and put in the chat window again. Okay, there you have it. So we have the plot window now. The next part of the thing is to say what to plot. it means that we need to include in the start function a vector, an array, to store the position of the center of the center of mass of the nucleus. This is done this way here. It's in blue here. See, the first line, I create a 3D array to store the current nucleus center of mass. The second array stores the previous position of the nucleus center of mass. In the third array, I store how much the nucleus has moved from one Monte Carlo step to another. Okay, I have to do that for one reason that I'm going to explain later. Let me copy these lines again. And put in the control copy. Here you have, okay, three arrays to store positions. The last line here, the loop over the cell type CITO. No, not CITO. I think I wrote the wrong name there. It should be nucleus, but anyway, it was going to work here anyway. It's the where I store the initial position of the center of mass of the cell here. Okay, it could be nucleus here. It doesn't matter. So we have the start function. Everything is 0, but I save the old position as the current position of the center of mass, where the cell was created, actually. Okay? Now I'm going to do step function. In the step function, I have to update this nucleus center of mass position. So I, again, each time here that enters in the step function, he enters in this loop here, where I have I use the center of mass plugin here, X-con, Y-con, take from the plugin center of mass the position of the cell type nucleus and calculate the difference between the current position and the old position here. Oops. The old position here. This is this displacement of the center of mass from the previous Monte Carlo step and this one. Okay. Then I save it in the old position to be used in the next time that the steppable is called, the actual position of the center of mass. Did you follow? This thing? I mean, I make the displacement between now and before,
I think Gilberto has some connection problem. I'm trying to call him now.
Give us a minute. We've actually had remarkably good luck with the internet so far, so I shouldn't say that too loudly. If there are any questions in the meantime that people would like to have answered, go ahead. We'll get Gilberto back as soon as possible. If he can't reconnect, we have options, backup options. We appreciate your patience.
So Gilberto asked me if I can go ahead and present where from where he stopped.
Okay. There
me
was a
share my
question about, please tell the people which exercise file you're working from now.
Exercise file. The exercise file is. Let me check here. The name is okay. Module 3.3, and the file is cellmig2d plot rec, which means plotting data and recording data.
Could you paste the name of the file into the chat just so that people know exactly which one it is?
Yes.
Yes.
So this is the folder, and inside the folder, the simulation is called cellmig2d. Inside this other folder, you must find the CC3D file, which you can open in your Reddit.
Okay, Pedro, if you want to take over, that's great. Thank you very much for stepping up and
Okay. I am actually showing a spoiler, and I don't remember exactly where
so we're seeing
Gilberto stopped.
there we go. Okay.
I think this is too small. There we go. And where did he stop? I don't remember. Okay, so here we have a piece of code here, nucleus center of mass, and which is basically initializing the arrays that we are going to use to plot data. And after that, in the step,
I'm sorry, Pedro, for some reason, the top of your screen is cut off in the
top of
display.
screen.
In your slides, they're sized. That's okay. If you go to display.
Okay.
No,
it's a problem in the format here. Maybe some version is incorrect. Now is it good now?
That looks perfect. Thank you.
Okay. And in step, okay, in the function step inside steppables class, you need to update your current position of nucleus based on the size of the step you made. Okay, you could plot your position directly, but there's a problem. Since we have a lattice with periodic boundary conditions, when the cell traverses one side, it will appear in the other side. So you will have your graph will be cut. You Can you visualize it? The cell will stop at a point and show up at another point. So if you just plot directly the position of the cell, this will happen. That's why we update the current position with the step we made in the previous step. Okay, the displacement of the nucleus in the previous step. Are you with me here? Let's see the chat. Unfortunately, I cannot see the chat and present at the same time. No, I can. I'm seeing here. Okay. Did you follow until here? Is your code exactly as Gilberto's code? Okay. Do you need me to copy and paste some part of the code for you? Please. Okay, so I will copy and paste from line 48 to line 72 of the slide I'm showing right now. Okay. So I don't have his code loaded yet. Here. And steppables. It doesn't show the line. I'll have to download the simulation as well. Is it plotting the trajectory with PVC correction? What is PVC? I'm not sure.
Periodic boundary conditions, which is exactly what you're trying to do here.
Yeah, no, when you have the periodic boundary condition and you plot your point, it doesn't correct by the teleportation thing, you know. Your cell disappear here and appear there, but the plot doesn't correct. We have to do this by hand. That's why this code is a bit larger than you thought. So please understand that I was not aware. I was going to substitute Gilberto now, so. And I said from 48 to 72. What's happening? I cannot copy and paste. Maybe I have to close the windows first. I don't know why.
Gilberto, do you want to continue or do you want Pedro to continue?
I cannot copy and paste.
Yes.
Cecily, can you download the simulation from?
Gilberto is back, I think.
Gilberto is
Pedro,
back?
do you want
Okay.
me to take over? My apologies, guy. My internet connection here failed for minutes and I was disconnected from everything. But now I can continue from.
I just explained why we are plotting the nucleus coordinates by calculating the displacement and not directly.
Mhm.
because the Compartmental does not correct the fact that we have periodic boundary conditions, and we need to save the displacement and correct it.
Okay,
So they
I will-
already know that.
Okay, I will share my screen here. Sorry, share screen two. Let me go there. So you were here.
I think you're not sharing your screen or yet.
No?
No, I'm not seeing your screen at least.
No, it's okay. I think it's there.
Ah,
Gilberto, you were okay. You were sharing.
okay. So let me see where did you
There we
start?
go.
Next,
Here? This part?
yes,
This
yes,
is-
this slide.
Okay, so as Pedro must told you, this part only takes into account of the fact that we are moving in an infinite plane, but we see only a part of it. So when the cell moves outside of the grid, we have to put it back in the other side of the grid. So if this displacer is bigger than certain amount, I have to discount that amount or increase that amount to the displacement to put the cell back in the window that you can see inside the simulation. Okay, this does everybody have part of it already? The code, the code. Did you cut and paste here in the chat window, Pedro? This part?
Yes, yes, just did it. People are
Okay.
using right now.
So the last line is here. You update the current displacement, summing the displacement that you have calculated and corrected in the lines above. After you that, you can add the displacement and that plot window, the graph window you have created in the start function. So you go to the CC3D Python scientific plots, add data points here, and add these lines to plot the actual position of the nucleus center of mass. So here I will copy these lines again. Control copy, Control V is that. And then after you do that, include this line, you are ready to go again. Save it. Every modifications you have done. Click on the CC3D logo, and the player will open with the graphical window aside of this main window, the cell field. Sure. Put it to run, and you have something like that. Sure. Let me put it to run here. Open CCD. since my network now is working. Here. File open simulation, recording plotting, CC3D. Right. See? Then you have the position of the nucleus being plotted in the graph trajectory window. Let's wait a little bit to your simulation works over there. Okay, so this is the end of this. Oh, no. There is an extra part of this, this lecture, this tutorial. That is now we need, as I told you before, also we have to export this data to be able to work with the data later. So how do you do that? You right-click on the plot window, and these options here will open. A drop-down menu will open here with the export option here. You click in this export option, and you have here the second window open, being open. And it asks you the export format. You want your data that is plotted in this window be saved. So you choose whatever you want here. Matplotlib, blah, 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 image, or however. You can save this window as a window, GIF, PNG or TIFF. But we wanted CSV, comma separated value from plot data. It's an Excel file like that. So you choose this one, choose what the separator you want for the data, comma. You can choose another option here, for example, tab separator between data, and click export. As soon as you do that, a file will be saved in your project folder here, okay? And this, actually, sorry, this window will be open to naming the file you want to save. For example, I am saving here as with the name nuc trash. It's a CSV file. Then save it. The saved file has this content: 2 columns, X center of mass, Y center of mass. And now you can do whatever treatment of data you want with those data. To calculate the mean displacement, the mean square displaced, to calculate the speed, the velocity autocorrelation function from this data here, because you know the timing interval, time in terms of Monte Carlo step that this data is being saved. It's saved at each Monte Carlo steps, and the time interval is 1, for example. Okay, and that's it. You have the data saved and the window with your simulation to be observed during on demand, let's say, on the fly, on running. Okay, this ends this part of the talk. The 2 and 3 main goals that I proposed from the beginning. The next one, can I go forward? Let's see in the poll. This one is the last one. I'm going to. Okay, 72% is doing the exercise. Okay. The next exercise will be presented by Pedro. This is a very nice simulation he did. And in this simulation, he will extend the previous one, the first one, the single cell simulation one, to include the multiple cells. Okay? Now, Pedro, take over.
Okay,
Thank you, guys.
thanks.
I will stop sharing here.
Yes, okay. I'll activate my camera. Just so you guys know my face. Let's here, start video. And there we go. Perfect. So I am a student of Gilberto. Gilberto, I am a graduate student, and we work with cell crawling. We work with isotropic conditions, which means no chemotaxis, no fields, and with anisotropic conditions as well. So maybe we can show you later if you have time. But for now, I'm gonna teach you how to build this simulation with multicell. So using this simulation, how can we introduce more cells? Okay. First question, what happens if we only create more cells with no caution? What do you think would happen if we only create more cells? Let's see what you think. Am I screen sharing?
You're not screen sharing yet. No.
Okay. Now I am.
Yes.
Perfect.
Okay. They will merge. Nice. You are too good, actually. So this is what will happen. They will bump into each other, yes, but they would bump anyway. So this is a cut from a simulation, and the nucleus are merging, the cytoplasm are merging. So this doesn't make any sense. This video doesn't seem right for you, right? Myofiber, yes, could be a myofiber, yes. But the nuclei wouldn't merge, right? So this way, I think you couldn't construct even a myofiber because the nuclei are separated. No, they do not. So this is a wrong simulation. Okay. Okay, Suvik. What do we want to happen? We want this. We want the cells to crawl individually. Yes, they will bump into each other, but they won't merge. Okay, this is what we want. But they are crawling individually. Yes, we can change that. We can change that with contact energies, but simply adding more cells, we won't reach the simulation we want. Okay. So first of all, how to achieve this? We need to build our cells in a way that the compartments that constitute the cell belongs to something that is different from the other cell, which is built with the same compartments. Multinucleate giant cells in TB. I don't know that. I don't know about that, but if you can share later, we'll be fine. And more than that, we need that those compartments inside the cell behave differently than compartments from different cell, you know? So for example, here we have a set of cells that I'm going to refer as a cluster now. So we have the cluster 1 composed by compartments 1, 2, and 3. And we have the cluster 2 composed by the compartments 4, 5, and 6. So what do we want? We want that the compartment 4 to have a to have an interaction with the cell, with the third cell, different from the interaction it has with the fifth cell, even if the third cell and the fifth cell are of the same type. You know, so we need to some way make those compartments belong to a cluster, to something new. And after we've done that, we need to give different energies for compartments that are interacting inside a cluster and compartments that are interacting outside the cluster. So there are two types of energies we are going to use now. We are going to use the contact energy, which you already know, and we are going to use the contact internal energy as well. So the contact internal are the energies of the compartments that are interacting inside the cell, and the contact will be the energies that will mediate the contact of compartments from different cells. Okay, so the Hamiltonian will be a bit different. It will use the J internal for same cluster, for compartments of the same cluster, and it will use the J external when the clusters are different. Okay, I bet it's clear for you. So first of all, let's start with the most basic simulation you have, which is the CellMig 2D. Open the CellMig 2D folder, which you can download from the drive, and load the CC3D project onto Edit. So this should be your simulation, CellMig 2D. Open it and load the CC3D file onto Edit. Once you've done that, your simulation should be just like mine here on Edit, and stay with the XML file opened. Let's make it a poll. You just need to load the CC3D project inside the CellMig 2D file folder. Okay, you're good. In the XML, delete the uniform initializer steppable. So go to the end of the steppable, okay, here, and delete the XML, and delete all the steppable module here. It's called uniform initializer. We won't use it because we want to draw the cells by hand, okay? We will draw the cells using Python after. So we are not using it right now. In Python, you can draw the cell in the shape you like. It's absolute free. So it's also interesting that we teach you how to do this at some time, right? So delete everything. No, not everything, just the uniform initializer. And set the Chemotaxis lambda to minus 150. So you go to the chat is. So you go to here. It's line 60. And you change the lambda to minus 150. It will make this force stronger, okay? Because the lambda regulates how strong the lamel will invade the medium. So when you increase in modulus, when you change it to further negative values, you make this force stronger, okay? You increase the probability that a lamel pixel will copy over a medium pixel. And after you've done that, I want you to start a new contact plugin, which is called the contact internal, but don't do it still, okay? Don't do it yet. I will first explain how it's done, and then I will copy and paste the code to you. So it will be faster this way. So what we want to do? We want to introduce the contact internal plugin and also the contact plugin, and set the contact internal energies to be different from that from the contact plugin, so we can control how they interact when the compartments belong to different clusters. So your code will look like this. You have the contact plugin with some energies and the contact internal plugin. In the contact internal, you are basically saying how the compartments will behave inside the cluster, and outside the cluster on the plugin contact. I am gonna copy and paste it for you. But I think you could do it by your own if you needed. Because it's an option available in the. I cannot copy and paste large things, so I will have to copy and paste by pieces. So this is the first piece, the contact. Notice that the energies are different. So those energies are different because we adjusted them. And the other piece of code is the contact internal, which I also sent to you. So your code should look like my code, which is in the right side of the screen. It's a really difficult job to adjust those energies. Did you pass the code? Yes, I passed it directly to Giuliano. I'm sorry, guys. I passed it to. I always committed a mistake. Always. So here's the first one. And here's the second one. Sorry, guys. Okay. Are you good? I'm going to continue now. What did you change before contact internal? Before the contact internal change, I deleted the uniform initializer steppable on the end of the XML file because we are going to draw the cells using Python. So at first I did this, and I also need to do this in my simulation here. I'm sorry, forgot that. Here. So it should look like this. And save. How did you select these contact energies? I tested very hard many types, many combinations. It's basically a parameter scan that I did until I got the format I wanted, and I mean the shape of the cell I wanted, and the speed I wanted to look, just visual, you know. But it's hard work. If you wanted to do this, it would be from scratch. It would be hard work. Contact internal is for energy between compartments of a single cell. Yes, which I am calling cluster. This cell, biologically speaking, we are calling cluster in CompuCell terms. And contact energy is for between different cells, which would be different clusters. Yes, that's it. So now I will proceed. And here. So now we need to create the compartments. Okay. What did we do until now? We set the energies, just the energies, and deleted the cell creation. Now we need to create the cell. So go to steppables. I mean cell mig 2D steppables.py, and go to your start function and give a new line there. And we are adding a piece of code to create each cell. We need 6 cells. So I am building 6 cells. And each of those cells must have a lamel, a cyto, I mean lamellipodium, cyto, and nucleus. Okay. So this piece of code is doing exactly that. And then I need to assign a set of lamel, cyto, and nucleus to a cluster. Okay, these are the 2 things I'm doing here. So look at the code now. I'm gonna pass and copy and paste after, but pay attention here. So first of all, I am setting our number of cells to 6. This is the maximum here in the way the simulation was done. The 6 cells is the maximum. And I am also defining some lists and a matrix of X and Y coordinates of all our cells. 10 and 50 are the coordinates of the first cell. The first cell to be created will have this set of coordinates here. The other cells will be created. They will be created on the side of the previous one. So we introduce the first coordinate, and then we change it. We give it an append to incorporate the other coordinates of the other cells. So to create all the cells with all the compartments, we do this loop over our n cells, and we use the command pots create cell to create a cell, a new cell. We don't know yet what it would be, but then we say the type of the cell will be cyto. So we are creating a cell of the cyto type. Okay, and then we give an append. We give an append of this cell in the list of the cyt, in the cyt list, which is the list of the cyto compartments. Okay, then we do the same thing for lamellipodium and for nucleus. So create, assign a type, and put it into a list. After that, we need to tell that this lamellipodium and this nucleus belong to the same cluster as that situ here. So we will use this function, reassign cluster ID, and take the number, the ID, the cluster ID of the cell seat, which is the situ cell, and pass it into the cell lum, which is the lamellipodium cell. And we do this, we do the same thing for the cell nucleus. And then we append a new set of coordinates inside our XY CM matrix. Okay, we have a chat. Let's see. Yes, exactly what James said. So I'm gonna copy and paste. I will take it from my simulation here. So it's all this piece of code here. I'll try to, yes, I can paste on the chat. And I'm gonna paste as well on my simulation here. So I am doing with you. So if some bug happens to me, it will probably happen to you as well, and we will be synchronized. So copy and paste. Did you understand? Code not here again. I'm sending only to Ajay. I forgot this. I always forget this to send to everyone. Okay, so this is the code. Copy and paste on the start function. Okay, so it should look like exactly as my simulation here on the right. Obviously not, yes, exactly like my simulation. And then you need to start a poll. Relaunch. There we go. So if you don't understand some part of the code, please ask on the chat, okay? Because you are not programming directly, so make sure you understand at least. And if you need help, please comment on the chat as well. Cells are not appearing. Yes, not yet. Explain line 30. Line 30, okay. So I have a matrix of X and Y coordinates of my cell, of all my cells, okay? So I have 6 cells. How will my matrix look like? I'm sorry, I'm using paint, but you know, whatever. I have 6 cells. So here's the first cell, the second one, the third one, the fourth one, the 6th one, the 5th one, etc. And here I will have a sublist of X and Y coordinates of the first cell, X and Y coordinates of the second cell, etc. Okay? So now look at my code. I introduced the first set of coordinates for the first cell. When I pass through this loop once, I have to append to this matrix the coordinates of the next cell and so on. So did you understand that? So to calculate the next position, I just do a plus 20 increment here on the X coordinate. So I just so I will create this other cell 20 pixels on the right of the previous cell. Okay, this is exactly what this line is doing. Did you understand? Got it. Nice. Okay, let's get back to the. So I think you all done that. Now we are going to create the cell effectively. I mean, we already created in the Compartmental software, but we didn't draw it. We didn't paint the cell in the lattice per se. And we didn't do it using the initializer steppable as well. So we are going to do this programming with Python. So the same thing, I'm gonna copy and paste the code for you, but please pay attention and understand what I'm doing here. First, I need my initial condition to be just like this. It's here I'm showing 4, but it will be 6 cells, okay? And each cell, each of these giant rectangles there are composed by 3 squares. The lowest square is the cyto, on the middle is the nucleus, and on the top is the lamel, okay? Lamellipodium. So I want to create it this way. To do this, I have this other piece of code here, okay? Same thing. Stay with me, try to understand, and then I copy and paste you. So first of all, I am introducing an object and a point object from Compartmental, which basically represents a pixel in our lattice, okay? And we need to create all our n cells. So first of all, I'm doing a loop here over all n cells. And then inside this loop, other 2 loops over x coordinate and y coordinates, okay, from -3 to 3. So basically this is a 6 per 6 square, no, 6 by 6 pixels. And we assign the x and y coordinates to our point PT. And we use this command cellfield.set to say, in this point PT, we want to have a cell of the cytotype and of the cluster ID NC. And the same thing for the nucleus and for the lamel. Okay, so here I am assigning this point PT to the nucleus, to a nucleus that belong to the cluster ID NC. Same thing for lamel. The only difference between the 3 is that I am increasing the y coordinate, so I create one above the other. Is it clear? If it's not clear, please please tell me on the chat. Now what I'm going to do is I'm going to copy. Where is it? It's not here, it's here. So this is our piece of code that I'm sending to you now. And I'm also pasting it in my simulation. No, again. What again? Again, no. What again? Other piece of code? Again. Yes. I think it automatically set to send to someone because I was sending to everyone before. I think it's some bug. Where is the NC list where the values are getting updated? Right, good question. So look here at my code. So the NC starts with this 4 here, okay? When those 3 cells are being created, they are created with the same NC index, okay? So whenever I make another loop over NC, I will always be getting those 3 cells of the same cluster, because from the beginning they were assigned to the cluster ID NC. I'm getting a strange error for the all codes in module 3. Unrecognized arguments. Can someone help him? I don't know if I can. I shouldn't stop to solve this problem. So someone please try helping Suvik. Okay. Now I'm gonna make a poll here. Okay. Your code should look just like mine by now. Okay, so only 2 people need more time. All the rest are done, so it's done, I mean. So I'm gonna move forward. Okay, so we draw the cells. Now, the last thing we need to do, and this is the last thing, we need to set other volumes for our cells. And we need to do this because we need smaller cells so they can fit in a more beautiful way in the lattice. And we also, and I am also decreasing the size of the nucleus, so it decreases the chance that the nucleus would break up the cell. So this is a way I found to solve that issue. So first of all, you can reduce the size of the nucleus so the cell don't break up, because with a large nucleus it has much more inertia. Obviously, you can't talk about inertia in pots, but I will leave it as this. And also fixing those energies, you can also do this. Okay, so by now you can run just like Forest. So where is here? Let me open my CC3D and show you how it goes. It's not opening. Ah, it is. No. Right here. So I click a step. Okay, some people don't know about this. I click a step first. I don't hit run. Okay, I don't give a play. I click a step so I can see the initial condition. Okay, I always do this. Okay, this is my simulation. And I also give 2 or 3 more steps just to make sure, and then give it a play. And I am unlucky, one of the nucleus disappear. Let me run again. And it's working, at least for me. Let's see the chat. Tá. Beleza. Could you do this? Mine looks the exact same as yours, except one lamel completely separates. Mine are stuck together. Yes, so probably your energies are not the same as mine. Okay, for all 6. Yes. If it's for all 6 cells, probably your energies are different from mine. So let me get back to my energies and show you the values here. Probably your values are different. Please check it out. Mine are all breaking. Check the energies. Check if contact internal, if it's contact internal here and contact over here. Check the neighbor order as well. Must be 4 in both of them. Okay, must be neighbor order 4. So C2 15, C2 22, and it's different. It's exactly the same of mine. I didn't set my lambda to minus 150. Yeah, this shouldn't be a problem. Did you check if it's contact internal and contact? Because all I did is exactly what I said. So I made it live so you could see. It was the lambda? Oh, okay, okay. Nice, nice. So Paulo had the issue and also Joseph. Mine was minus 500. Yes. What happens when it's minus 500? The lamellipodium will try to invade the medium as much as it can, right? So it will disattach from the C2 completely. I misspaced it, okay? Missing the second lamel and nucleus, okay. Nucleus energy statements. Okay, so I can copy and paste again. No worries. And now I'm sending to everyone this and this. With this presentation, we are able to perform cell division in the same way as, yes, you can. Everything is allowed, even with contact internal. But, many buts, you need to make sure your created cells belong to the same cluster. So every time you create a new cell, you must use that command cluster reassign cluster ID. Okay? You must do this every time you create a new cell. If you want that cell to belong to a cluster, you need to say it. So with this, guys, you can make something you couldn't do before, which is, for example, let's say you have two groups of 10 people. One group love people love between them, the other same thing, but the two groups hate each other, and they segregate. They are all of them are people, right? So they are like cells of the same type, but they hate each other. They love each other in the same group, but they hate each other. This very, very simple model, you need to, if you want the cells to be of the same type, right? You need to tell me the error, Tahrim. You need to use contact internal and contact. For all, now I understood your question. Sorry. If you want to perform a cell division, yes, you would have to divide all three compartments, or you could divide only the nucleus and make the nucleus create the other cells. It's also another option. And Juliana said there's a cell division for clusters. Perfect. Okay. Ajay and Tahrim, if you want to go to a breakout room. Okay. So I will proceed with the presentation now. I hope everyone at least understood what was done. Okay. So now you have an exercise to do. You have a homework. a homework. The homework is to create a steering panel. So a steering panel is something very key here, okay? Because adjusting those energies is very, very hard work. You need to, you go to your file and you change here, here, and there, and then you run the simulation, then you go back, and then you change here, 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 and there. And this is very difficult, mainly because all those energies, they form like a system, a multivariable system, where you change one energy and this effect will change another effect in your simulation. So it's like trying to tuning a bad guitar, a really bad guitar. You probably know what I mean. You tune this chord here and this string here, and the other string get out of tune, and vice versa, you know? So it's very difficult. With the steering panel, it will be much more easy, because you will have those buttons you can adjust, and then with the simulation running, you don't even need to pause the simulation. You can adjust your energies during the simulation, and also you can adjust the Lamel target volume, the Lamel lambda volume. You can adjust everything, okay? You can adjust volumes, energies, lambdas, and also the chemical lambda. Okay, so this is your homework. We have a demo for that. So if you installed CC3D in your PC, you have a demo called steering panel. So you can base yourself in this demo to make this homework. And this is very useful for you. We also have the result. So the simulation result is also uploaded on the drive. So if you want to jump straight ahead to the simulation, see how it's done, it's there. Right. So I'm actually gonna show you this code now. Let me just pause the simulation and open the simulation with the steering panel. So where is it? I think it's downloads. Yes, steering here. So use the single cell simulation, please. You don't want to suffer extra. So this is the steering panel. Okay, are you with me? So the steering panel is an amazing piece of work, where you just change these guys here from up to down values here, and while the simulation is running, you'll see the effect. So I'll hit play here, and I will increase lambda volume, and boom, look at the lamellipodium now. So now you have a completely different cell, right? So the shape changed, everything changed, the speed of the cell changed. So I can reduce the C2, for example. Let me reduce much more. I can make an egg. So if I do this, for example, and let's make this. So probably I will make an egg doing this, not quite. So let's change the chemical lambda again and see what happens. So the cell broke up, but it catches right away. So broke up again. And you can play a lot with it. So you can vary your parameters using the steering panel. It's much more easier. let's try other parameters. And I mean, you can put all the energies here on the steering panel, right? So look, bigger C2. You can make a extremely elongated cell, but it breaks up. You can avoid the cell breaking up if you add some more how can I say some more plugins, like that plugin that how it's called, let me see, plugins don't remember, it's not here, but you can add a plugin that avoids cells from breaking up. You can do a lot of things and put everything on the steering panel. Just one more thing, be careful because steering panel consumes processing time. Okay, so if you are trying to do a really fast simulation, you probably will want to vary your parameters outside the yes, of course. I almost forgot. Let's open here. Okay. And here. Ooh. I closed the window in the left side here. Let's go to the notepad. Okay, so here I have the simulation with the steering panel, and what you want to add here, it's the following. First, every class you create on Steppable should be called on your file .py, your .py file, not Steppable. You know, that third file you don't hear much about. So you have the XML, the Steppable, py, and the py. Okay, so in the py you always call the classes you are introducing. For example, here in steering panel, I am introducing here a class steering steppable. Okay, so this class is being called here on the py file. So you will have register steppable, steering steppable here, and also calling in the import head. Okay, so make sure you always call your class. In the XML, if you want, for example, to put in your steering panel the volumes, you have to delete the volumes here in the plugin on XML, because if you set some volume in the XML, it will overwrite the volume you are setting on the parameter scan. This volume is higher on the hierarchy of the values, you know. So if you want to add a value in, if you want to change a value in the steering panel, you need to delete from your volumes here from the XML. Okay, if you want to change an energy, so it's not done here, but I can show you how. you need to, I don't know this, I don't remember the syntax exactly, but you need to introduce an ID for a specific energy. So if I want to change medium nucleus energy, I need to tell to make an ID, for example, like ID equals nuc maj energy, something like that. And in the simulation, I can access this energy by calling it nuc med energy. Let's see if I find it in some simulation here. No. Instantiate the steering panel. Ah, yes. Oh, I cannot use my tweeted because I closed the left window. How can I? No. I'll try resetting the. CC 3D project show project panel. Ah, okay. Or resetting Twitch. Okay. Here. So if I wanted to do it from scratch, let's say, I would be here in Steppables. And let us open a clear space here. And I would go here. So CC3D project. No, CC3D Python. And you go to steering panel and set up a steering panel. Where are the classes? It should create the class as well.
Hey, Pedro, this is potentially a pretty detailed bit of technical work for how to do this. How do you feel about can grab the link in the documentation for how to do this for now, for those who might be interested, and then we can go through it a bit more rigorously in a later session this week.
Yes, I agree.
Great. Okay, I'll grab that link. Yeah, so you don't have to worry about it.
Yeah. So there's a demo on the demos inside the installation folder of Compartmental that you can check out. We also have uploaded the simulation with steering panel. And also check out the documentation Giuliano posted on the chat, which is the manual. Okay, so thanks for your patience.
Okay, thank you very much. I mean, these are sophisticated things, and so it's fine to, if you didn't follow all of that, don't worry, you'll get to see it again. I appreciate Pedro being willing to step in for Gilberto when there was a technical problem. So are there other questions about this particular module before we close up for the day? There's a question.
Yeah, I have a quick question. This might be technical. So so I looked at the cheat sheet, but I didn't saw is that the opposite n. So those the properties for self are is it built-in attributes?
Which attributes are you talking about?
I'm talking about like the cell mic to the stepable dot pi. So if you look at the self dot POSN.
Cell
yeah.
dot.
Yeah. Like DS1N and OPOSN. Yeah.
Okay, here.
Yeah. Are
Here,
these?
okay.
Yes. Are these attribute are built-in? Because I didn't find it in the cheat sheet for the attributes.
So this here is Python programmation. It's not it has nothing to do with the Compartmental itself. So this is generic. You are creating a variable that belongs to the entire class. So when it has the self dot before, it means you can use it between among all the functions inside the same class. So I could create another cell like self Pedro here and tell it is nice. And then I take this variable here and I jump to another function and I can print this variable with no problem. Okay, so those things here are just created things here in the simulation that we use to pass from one function to the other. So in this case, why are we using the self dot some name, right? Some variable in this simulation here. It's because we need to define it. We need to define them in the beginning of the simulation, in the start. So, and then use them after in the step. So in order to do this, we define them as self variables.
Okay, thank you.
Multinucleated cells, like giant multinucleated. How would we? Probably we would have to create like different nucleus types. And if you want a variable number of nucleus, which is very real, right? So for example, muscle cells have a variety of numbers of nuclei. So if you want to do this, probably you would want to I don't know how to do it actually. Not right now, but It can be extended to multinucleated. Yes. Yes. Yes, you can. If you want a variable number of nuclei, it's gonna be much more difficult, I think.
No, I think it'll work. You can create compartments if the nuclei don't adhere to each other.
Ah, yes.
You can create as many as you want of the same subcellular type within a new cell. It'll work fine.
Yes. Yeah. Ah, yes.
In fact, the simulation you have will do it if you like.
Let's see if there's another question here.
I'm sorry, if you use the contact energy at the same time adhesion flex, I mean by introducing some cations and these kind of things in order to define adhesion energy. So which one would be in the effect? I mean, do they work at the same time, these two plugins?
I didn't try this, so I cannot answer the question. Sorry, I didn't try. I don't know what would happen. Maybe James.
Sorry, could you say that again, please? I didn't catch that.
Is it possible to use the contact plugin at the same time with adhesion flex plugin in order to introduce contact energies?
You probably would want to just use adhesion flex. You don't really need both. And I don't know, TJ, if they can run together.
They can, and it would have an additive effect, but
That's what I expected.
yeah, but it would be a major redundancy and also pretty unnecessarily expensive computationally, because anything, as James said, anything you could do with the contact plugin, you could do in adhesion flex.
Wow, yeah, thank you.
So I appreciate people's patience today. We got through there. You covered a lot of material today. I just wanted to say that we'll reorganize things a little bit so that the lecture that I was going to give now, which is a very, very gentle introduction to subcellular network modeling, we'll do tomorrow morning. And then we will continue with the hackathon teaming exercise, and Joshua will show you how to import SPML models into CompuCell. And then we'll continue with antimony tellurium modeling in the rest of the day tomorrow. And I don't know exactly how far we'll get, whether we'll get to the MobBoss integration tomorrow or whether that will be on Friday morning. But we can, the amount of time we spend on the hackathon teaming is a little bit up to you. I don't have, there are going to be 4 people presenting their project ideas. I want to give time for people to ask questions and talk together. We can create breakout rooms for teams to try to meet with each other and come up with ideas. So those were the main things that I wanted to discuss today. If there are any questions or comments, please feel free to talk to us. And that would be, that's really the main set of things that I had. So if there are any questions, people are free to unmute and talk about any issues that they're concerned about.
Will we be starting at the same time tomorrow?
Yes, we'll start at the regular time of 10:30, 10 o'clock for setup and discussion, and then 10:30 for the first formal module. And that formal, sorry, go ahead.
I was just wondering if there was any reason why we couldn't start in the sort of early questioning time, since that typically we haven't had many questions. Just
well,
to kind of help get things fitted in.
The only problem with that, I understand why you say that, and I'm sympathetic to that. The only problem is that but since the official start time is 10:30, if we start early and people don't know, they miss the beginning of the lecture.
Yeah, that's fair.
And I understand that there's no perfect time zone. We have people in India who are staying up all night for these lectures. We have people in Europe for whom this time now in the evening is late. But we also have people on the West Coast of the United States who have to get up at 7:30 in the morning for a 10. I mean, where the lecture starts at 7:30 in the morning for a 10:30 call for us. And so if we push it, there's no perfect. Yes, poor Suvik, we're very appreciative of you staying up all night for this. There's no ideal time. And so when we scheduled it starting at 10:30, we were trying to make it and ending officially at 5:30. The goal was to try to make it manageable as possible for everyone in the US and people in Europe. Then so originally we didn't have that 10 to 10:30 booked at all. But we thought that since we would be there, it would be an opportunity for people to ask questions. So I understand your question, but I'm reluctant to change the schedule today because of Gilberto's internet issue and a few other things. We had a little bit of a schedule slip, but I think we have to stick with the original schedule. I know there are people who are planning on attending who wouldn't know between now and tomorrow about the change of time. So I apologize for that. Is there anything else? Yes, I understand it's very late in Europe. So I appreciate people sticking around. Again, if there are no questions, you don't have to stick around. But it is an opportunity to talk more openly. Okay. Okay. I guess if there are no other questions or comments, I'll look forward to seeing you all tomorrow. If the people from the class, the instructors, could stay on for a few minutes, I'd appreciate it. Everybody else, will see you tomorrow morning, whether at 10 or 10:30.